This is the third day today of the marathon revision session that we have arranged for you to help you clear your Part-1 exam. And I am Dr. Chitra, your mentor at Study Medic and Study MRCOG. And today I would try to make embryology and genetics a little bit easy for you, explain the important concepts that are required and how to solve SPAs in the exam. And we would be discussing about the important topics or the hot topics that are likely to appear in the exam. So if you have a look here, the weightage is relatively as an optional topic, because that one point that you get correct in the exam, that could be your savior point, and it would decide whether you are going to clear, like you're reaching the expected standard or you're not reaching the expected standard. So please do not leave any topic as an option. Each and every point is important. All right, so let us go ahead. We will first start with embryology. Now in embryology, the important topics are early development of the embryo. That means fertilization up to gastrulation. What about twinning? What is the basis, or how do you get formation of the various type of twins? How does the heart or the cardiovascular system, how does it develop? Renal and urogenital, because as obstetricians, we are mainly concerned with those two systems. And then sexual differentiation and disorders, or what was formerly called as intersex, and teratology. And we would be seeing, we would be solving important SBAs from the above topics. So, fertilization, we know it occurs in the ampulla of the fallopian tube within 24 hours of ovulation. This could come as an SBA. So the timeline in early development of the embryo is very important. Now, first, when you get fertilization, you get the zygote, which is one cell, and it is diploid because you're getting 23 chromosomes from the sperm, 23 chromosomes from the ovum. Now, first division occurs approximately at around 48 hours. And then this zygote starts rapidly dividing. And on day 3, you have a structure which is called as the morula, which has 16 cells. It is a solid structure. That means there is no cavity seen in this morula. Once the cavity forms, the morula is called as the blastocyst. And when you have the formation of the blastocyst, you have the cells divide into two parts. You have the cavity, you have the inner cell mass, and you have the outer cell mass, which develops the trophoblast, or that which develops the placenta. Moving ahead, day 6 to 7, implantation occurs. Implantation occurs in the uterus, usually in the posterior wall on the upper segment of the uterus. And this is very important because if there is some defect in transport of this morula from the tube to the uterus, if implantation occurs in the ampulla of the tube, you would be getting what is called as an ectopic pregnancy. Okay. Day 6 to 7, implantation occurs. By this time, this blastocyst is going on dividing. At this point of time, you have two layers of cells. It is a bilaminar disc. This I'm talking now about the inner cell mass, because this inner cell mass is the part which is going to develop into the fetus. So this is a bilaminar disc right now. And the types of cells that are there, they are the epiblast and the hypoblast. On day 14, an interesting thing happens, which is called as gastrulation begins. Okay, so it has nothing to do with the GI system, but it is called as gastrulation. That means formation of the 3 germ layers. And what is the triggering point for this? It is formation of the primitive streak. It starts at around day 14. That means in the beginning of the 3rd week, gastrulation starts. So you first have the primitive streak, then you have the node, the pit, and the groove. So what happens when gastrulation starts, or the 3 germ layers start? At this point of time, The embryo that is there, the laterality is defined. So you have the right side of the embryo, you have the left side of the embryo, the anterior part, the posterior part, caudal end, and the cephalic end. Okay, so that is established, and then you get formation of the three germ layers. Which are these three germ layers? The epiblast that is there, it starts proliferating rapidly, and then it forms endoderm, ectoderm, and mesoderm. Okay, so ectoderm is the surface layer. It is called as the surface layer. Mesoderm is the one that is there in between the ectoderm and the endoderm. Now, it is very important that you understand the types of tissues that are going to be formed from the ectoderm, mesoderm, and endoderm. Okay, so ectoderm, we know it is the outermost layer. So skin, epidermis, hair, nails, then whatever is exposed to the surface. All of these structures, they are going to form from the ectoderm. That is one. Obviously, there are exceptions to every rule. So adenohypophysis, which is not exposed externally, that is formed from the ectoderm, specifically a layer called as Rathke's pouch. And this Rathke's pouch, it develops from the oral ectoderm. Now mesoderm, M for middle, remember. So whichever structures are there in between the ectoderm and the endoderm, they are formed from the mesoderm. All your musculoskeletal system, the cardiovascular system, urogenital system. Urogenital system, remember, specifically from the intermediate mesoderm. And these two, they develop in very close contact with each other. So whenever you have disturbances in formation of the renal system, it is also associated with disturbances in the genital system, or you could say vice versa, like disturbances in formation of the genital system always look for any development anomalies in the renal system. Another exception to this, the adrenal cortex. Please remember, adrenal cortex develops from the mesoderm. Now endoderm means it is the innermost layer. So all the lining of the GI tract, the respiratory tract, then the urogenital, everything, all this innermost lining, it comes from the endoderm. So inside the tubes and all the lining, that is your endoderm. Now you have to know a little bit about the neural crest. So what is this neural crest? It is a type of special ectoderm. Your peripheral nervous system, then the pigment cells, endocrine. Remember, cortex develops from mesoderm, but medulla, it develops from neural crest cells. It's very important to remember. Okay. Neural crest. Also, the parafollicular C cells, they develop from the neural crest. And in the craniofacial system, your facial bones, jaw, all those things, they develop from the neural crest. So, in the exam, please remember adrenal medulla, it comes from the neural crest, whereas the cortex, it comes from the mesoderm. My way of remembering it is M is medulla, but medulla doesn't come from mesoderm. It is the opposite. So medulla with neural crest and cortex develops from the mesoderm. Another important thing to remember is about the bladder trigone. Bladder, we know, develops from the urogenital sinus. Urogenital sinus develops from the endoderm. But a part of the bladder trigone, that develops from the mesoderm or from the mesonephric duct. So these are important things or important points from this slide. So we already saw what are the structures that develop from the three germ cell layers. Something important about the notochord. So this notochord, it develops from the ectoderm, the midline mesoderm, and that induces formation of the neural plate. Now remember the neural tube, it closes at about 4 weeks. So if this doesn't close at 4 weeks, anterior closes at 21, the posterior neuropore closes at about 28. So if anterior doesn't close, that gives rise to anencephaly. If the posterior doesn't close, it gives rise to spina bifida. And you can sometimes, it is deficiency of folic acid which prevents closure of this neural tube. So giving folic acid preconceptionally, it is very important to give folic acid in the preconceptional period, because see, neural tube already closes by 4 weeks. If you start giving folic acid after the urine pregnancy test is positive, it will really not help you to prevent formation of neural tube defects. That is why preconceptional period, 3 months before the woman plans her pregnancy, it is very important to start folic acid. That helps to prevent neural tube defects. Now, you have Hox genes which regulate body axis patterning. So Hox genes are basically, these are master regulatory genes. You can call them as architects of the human body or organism, that whichever is developing. If these Hox genes, they do not work as they should be, then you will get all congenital anomalies or malformations that are there. So important points to remember about early embryology, that means fertilization to gastrulation. Remember, fertilization takes place in the ampulla of the fallopian tube. Something very important to remember again here, that ampulla is the site of where you get the most ectopic pregnancies. Implantation occurs on day 6 to 7 in the deciduum of the uterus. Gastrulation begins day 14 to 15 or at end of second week. Ectoderm, that is the outermost of the 3 germ layers, that forms the skin, central nervous system, and adrenal medulla. Mesoderm, or the middle layer, it forms bone, muscle, kidney, and heart, or basically all the systems that are there inside. And endoderm, which is the innermost layer, that forms the lining. So gastrointestinal lining, liver, and pancreas, all that comes from the endoderm. Adrenal medulla, remember, it comes from the neural crest or cells which are called as from specialized ectoderm. And the cortex, adrenal cortex, it comes from the mesoderm. Bladder trigone, rest of the bladder comes from urogenital sinus, but the trigone, it comes from the mesonephric duct. So normally we know mesonephric duct is something which regresses in females, but in the female also, importance of the mesonephric duct is there because that leads to formation of the bladder trigone in both males and in females. And another thing to remember, neural tube closes by 4 weeks. So if at all you want to prevent neural tube defects due to folic acid deficiency, it is important that you start giving folic acid in the preconceptional period. Now, coming to the hot topic of twinning and placentation. Okay, we know there are different types of twins: dizygotic, monozygotic, monochorionic, diamniotic, dizygotic, diamniotic, monochorionic, monoamniotic, and you also have conjoined twins. So, if you are not aware of the time zone when the splitting occurs and what time and what type of twins you are going to get, then it is something which will cause a lot of confusion. But I will try to explain it to you in a very simple manner. First thing you have to remember, there are 2 types of twins, monozygotic or dizygotic. Okay. So dizygotic twins are the commoner ones. About 2/3 of twins are normally these days, they are dizygotic. Now, what is the meaning of dizygotic? Means 2 zygotes are there. So there are 2 eggs which are fertilized by 2 sperms. Now, because there are 2 different entities, okay, 2 eggs are being fertilized by 2 different sperms. So basically, in one uterus, you are getting development of 2 fetuses. And because the egg and the sperm is different, always DCDA twins are by default, they are dichorionic, diamniotic. That means they do not share a placenta. It is never possible that dizygotic twins will share a placenta. They are anatomically different, anatomically and physiologically different from each other. Okay, so they are basically 2 separate pregnancies which are developing in one uterus, and they are also genetically distinct because they are not sharing any genetic material. Which are the women that are likely to get dizygotic pregnancy? So family history, IVF, advanced maternal age, And these days now, because many pregnancies are because of IVF, we are seeing a rise in the incidence of dizygotic twins. Monozygotic twins are the ones that are about 1/3. So they are very much rarer than dizygotic twins. Now here what happens is you have one egg and one sperm. So basically when fertilization is happening, there is only one egg and one sperm. And then afterwards, this due to any cause, there is splitting of the zygote that happens. It splits into two parts. Now it is basically one zygote that is being split up into two parts. So that is why monozygotic twins are always genetically identical. Okay. And chorionicity depends on the day of splitting. So depending on whether it happens early in pregnancy or later on, you get three types of twins. Now we already saw in early development that up to day 3, you have something which is called as the morula. In the morula, you have 16 cells, and these cells are not differentiated into the inner cell mass and the outer cell mass. So if splitting occurs, okay, between day 0 to day 3, you get formation of two complete pregnancies, okay, two different patterns of cells are there, and because the cells in the morula, they are totipotent cells, they can develop into the inner cell mass and the outer cell mass. Okay, so that is why if splitting occurs between day 0 to day 3, you get formation of two completely different pregnancies in a utero with two separate chorions and two separate amnions. Okay, but please remember, this is totally this you get dichorionic, diamniotic twins, but remember, these are monozygotic twins. They are not dizygotic. It is only because the splitting has occurred very early that you get formation of two placentae and two amnions. Now, DCDA twins are about 25 to 30% of monozygotic twins. How will you know whether this is a monochorionic or dizygotic? So if it is a DCDA twin, when you are doing the scan, you will get something, the lambda sign. Okay, where the membranes are being attached, you get the lambda sign. And this, the membrane that is there in between both the twins, that is a very thick membrane. And because there are two different placentae, this DCDA twin, it has very low risks. We will see what are the risks that are there in a MCDA twin. Okay, because basically they do not share the placenta. There are no there are no anastomosing channels between both the twins. So DCDA twins have comparatively lower risks than MCDA twins. Now, if splitting occurs on day 4 to 8, that is the time, remember, we already saw that the inner cell mass has already developed and the outer cell mass has also differentiated. That means the placenta has already started forming. And it is only the inner cell mass now that has split up. So you get formation of one placenta. And in between that, inside the inner cell mass has now divided into two. So you will get one placenta. You will get formation of the amniotic sac. So two amniotic sacs are there, and there are two different twins, that are two different babies developing, but they share a common placenta. So that is your monochorionic diamniotic. That means one placenta, two amniotic sacs. This is about 70% of monozygotic twins. Now on ultrasound, you will see the T sign where the membranes are being attached. And this membrane that is there, it is a very thin membrane. And the risk of TTTS, or twin-to-twin transfusion syndrome, is about 15%. This monochorionic diamniotic MCDA twins are at a very high risk of getting all complications of twins. Now, if splitting occurs after day 8, now remember, after day 8, chorion is already formed, the amnion is already formed. So you will get only splitting up of the inner cell mass, and even that splitting, it will be, so you will get two twins here, and there will be no dividing membrane, no amniotic no amnion is formed because it has already been formed and only the inner cell mass is splitting up. This is something very rare. MCMA twins is only about 1% of monozygotic twins. There you will not see any separating membrane on ultrasound. These twins are at very high risk of complications because of cord entanglement. Okay, and very high risk of intrauterine fetal deaths, and they have to be delivered very early at around 32 to 34 weeks. And you can also get trap sequence in these twins. Now, if the splitting occurs later on, after day 13, after the axis, that means day 13 means you already saw that the primitive streak is already there, gastrulation has already started. Then you get formation of conjoined twins. These are very rare conjoined twins because the splitting is incomplete. So placenta is formed, the amniotic membrane, everything has been formed, and partial separation of the inner cell mass. You can get them joined at the head, like it could be thoracic or cranial or lumbar at any point. But conjoined twins are something which are very rare. So that is all about how you get formation of dichorionic diamniotic, monochorionic diamniotic, and monochorionic monoamniotic and conjoined twins. These are all types of monozygotic twins. Please remember, dizygotic twins are always dichorionic diamniotic. So to revise once again, if the splitting occurs between day 0 to 3, you get DCDA twins, which is about 25 to 30%. If splitting occurs from day 4 to 5, you get MCDA twins, which are 70% or more common of monozygotic twins. Day 8 to 13, after the amnion is formed, you get formation of monochorionic monoamniotic twins, which are very rare, about 1%. After splitting after day 13, you get conjoined twins, which is something again very extremely rare. So which is the best time to assess chorionicity? So the best time is between 11 to 14 weeks, and it is best assessed done a transvaginal scan. Okay. So at the point where you get the insertion of the membranes, if you get a structure like this is called as the Y shape or lambda, then you can say that this is a DCDA pregnancy. Okay, this is DCDA. Otherwise, where the membranes are being attached and you get something like this, or what is called as the T sign, then this is, or the membrane is very thick, this is MC, monochorionic diamniotic. Okay. And in MCMA, no membrane is there. You will only get formation of the amnion and then you will get the placenta. Now for MCDA twins, the risk is very high. You get TTTS, that means twin-to-twin transfusion syndrome in about 15% of this MCDA twins. There is selective fetal growth restriction. That means one fetus shows fetal growth restriction. And then you can get TAPS, that is twin. It is a sequelae of treatment of TTTS. Oh, so anemia, polycythemia, sequelae is seen, like one twin is anemic, the other twin develops polycythemia. It is because it's a consequence of treatment of TTTS. Then what about MCMA twins? These are twins which have the highest risk of getting all complications. So cord entanglement, because there is no amnion, no amniotic cavity that is separating both of this, but you get formation of two cords. And these twins are moving. So this is your amniotic cavity and you have your two cords here forming and the attached fetuses. And there is no membrane. So these two twins are freely roaming around in this amniotic fluid and then the cord gets entangled. Okay. In this, you can get formation of trap sequence. That means there is twin reversed amniotic fluid. So in this trap sequence, what happens is there is one twin, which is the donor, that keeps on pumping blood into the other twin that becomes the recipient. So this donor twin, it becomes very small, and the recipient, it starts growing big. And in this recipient, after the trap sequence, there is no formation of the head or the heart, and you are very likely to lose both of these twins if you have formation of trap sequence. So to summarize, dizygotic twins are always diamniotic, dichorionic. Monozygotic twins chorionicity depends on the day of splitting. If it is between day 0 to 3, you get formation of DCDA twins. Day 4 to 8, you get monochorionic diamniotic, commonly called as MCDA twins. These are the commonest type of monozygotic twins. Splitting occurring between day 8 to 13, you get monochorionic, monoamniotic. If after day 13, you get conjoined twins. Lambda sign is seen in dichorionic, diamniotic, monozygotic twins. T sign is seen in monochorionic, diamniotic. Risk of TTTS is highest with monochorionic diamniotic twins, about 15%, and chorionicity determination is best done at 11 to 14 weeks, or when you are doing the dating scan. That is the best time to assess for chorionicity. All right, now next coming to development of the cardiac system. So fetal circulation is something that you have to be very much aware of. You have to know all the bypasses that are there in the fetal circulation, and what is the development of the fetal heart. So the heart is one of the earliest organs to start functioning. On the first time you can see the cardiac activity is at week 3. When the heart starts forming, basically you don't have the 4-chambered heart. It starts developing as a tube. Okay, it is a single tube, and then this tube develops and it undergoes rotation and all those things, and then you get formation of the 4 heart chambers. So this single tube, it forms by week 4. Now in this, you have formation of the truncus arteriosus. This truncus arteriosus, it separates and it gives rise to aorta and the pulmonary trunk. Now, if there is failure of septation of this truncus arteriosus, you get what is formed as the persistent truncus arteriosus. Then, okay, the parts that are there of this tube, you have the truncus arteriosus, you have the bulbus cordis, you have the primitive ventricle, primitive atrium, and you have the sinus venosus. So truncus arteriosus, it will give rise to the aorta and pulmonary trunk. Bulbus cordis, remember, it gives rise to the smooth parts of the ventricles. That means the right outflow tract that is formed from the bulbus cordis. The primitive ventricle, it gives rise to the trabeculated parts of the left and right ventricle. The primitive ventricle, it gives rise to the major part of the ventricle. The primitive atrium, it gives rise to the major parts or the trabeculated part of both the atria, right and left atrium. And the sinus venosus that is there, it forms the smooth part of the right atrium, superior vena cava, and the coronary sinus. Now coming to formation of the interatrial septum. Okay, remember in the fetal circulation, blood does not go into the lungs for oxygenation, basically because the lungs are not still functioning in the fetus. So the right ventricle, that is not pumping blood into the fetal lungs, it's not going into the lungs. Blood in the fetal circulation, it goes from the right side of the heart, it goes directly into the left side of the heart. Okay, excuse my drawing, I'm not very good at drawing. I will just draw basic diagrams for you. Okay, so if this is your heart, these are your 4 chambers. This is your right atrium. This is the left atrium. This is the right ventricle, and this is the left ventricle. So in fetal circulation, what happens is blood comes into the right atrium, and from the right atrium, it directly goes into the left atrium. From the left atrium through the mitral valves, it goes into the left ventricle, and from the left ventricle through the aorta, it is distributed to the rest of the fetus. Now, normally in an adult, blood doesn't flow from right atrium to left atrium. So there has to be some connection in the fetus for blood to go from the right atrium to the left atrium, right? That is called as the foramen ovale. Now, how does this foramen ovale develop. So when you have this development of the interatrial septum, it first begins as formation of the septum primum. This septum primum, it starts forming from the base or the apex, and it starts developing from the apex and it goes downwards. Now, when it is developing, initially there is some connection between the right atrium and the left atrium, and that is called as the um, foramen primum. Okay. Then there is another septum which develops to the right of the septum primum. This is called as the septum secundum. Eventually, both of these, they fuse with each other. Okay, the septum completely develops, and you have an opening in between, which is called as the foramen ovale. Now, the flow of blood, remember, in the fetal heart, it is basically from right to left. Okay, so blood goes from the right side to the left side. This foramen ovale, functional closure occurs at birth. So once the baby takes the first breath, what happens is pressures in the right side of the heart, they fall, and pressures in the left side of the heart, they increase. So blood is now no longer able to flow. It will not flow from, it always flows from high to low, right? So now after birth, what happens is in the right atrium, the pressure will fall. Pressure increases in the left atrium. So functionally, this foramen ovale closes at birth. Anatomical closure occurs a little bit later. Now, in about 25% of individuals, the foramen ovale, it remains patent and it doesn't close. Now, next coming, okay, so the other parts of this, the septum primum, the lower part of the septum primum, it forms from the endocardial cushions. So this endocardial cushion that is there, it forms the atrioventricular valves and the lower part of the septum primum. Now the ductus arteriosus that is there, it develops from the sixth aortic arch. It's a derivative of the sixth aortic arch. And what is the function of this ductus arteriosus? We saw that normally blood does not flow from right atrium to the left ventricle. But not all blood goes from the right atrium to the left ventricle, right atrium to left atrium. A little part of blood escapes and it goes into the right ventricle. But from the right ventricle, blood cannot go into the lungs, okay, for oxygenation, because lungs are, it's a very high pressure system. Blood cannot be forced into the lungs. So there has to be a way out. So that is why you get this ductus arteriosus, which takes all this blood that has by mistake, the blood which has come into the right ventricle, that goes into the descending aorta. Okay, so that is one of the other bypass that is there in the fetal circulation. Now this ductus arteriosus, just like foramen ovale, it is going to close after birth, because after birth blood is going to flow from the right ventricle. It is going to go into the lungs for oxygenation, and you really do not have any function of this ductus arteriosus. But it does not close immediately after birth. It closes between, functionally it closes 24 to 72 hours after birth. Okay, and the anatomical closure, it happens a little bit later at around 1 to 3 months. And the resultant, the structure that you get is called as the ligamentum arteriosum. Sometimes this ductus arteriosus, it fails to close, and you get what is called as a patent ductus arteriosus. So in preterm babies born before term, because this ductus arteriosus, it is supposed to close at term after the first breath of the baby. So whenever you have any preterms, in them you will have a patent ductus arteriosus. Rubella infection is another infective cause where you will have a patent ductus arteriosus. Now, wherever you have coarctation of aorta, or there are defects in the formation of the aorta, you have two types of defects. They are preductal or postductal. For your exam point, you really need not go into details of what is preductal and postductal. Okay. So to recall about development of the heart, remember heartbeats starts at week 3. Truncus arteriosus gives rise to the aorta and pulmonary trunk. Whenever there is failure of septation, you get a persistent truncus. The sinus venosus, it gives rise to the smooth part of the right atrium and the superior vena cava. Ductus arteriosus is a derivative of the sixth aortic arch. The foramen oval functional closure occurs at birth or at the first breath. Anatomical closure of foramen oval occurs at 3 months. Ductus arteriosus closes functionally 24 to 72 hours after birth. Anatomical closure at 2 to 3 weeks. After the ductus arteriosus closes anatomically, the remnant that is there, it is called as the ligamentum arteriosum. Which are babies that are at a high risk of developing patent ductus arteriosus? So preterm babies, rubella infections, and sometimes even at a high altitude, you get failure of closure of the ductus. Now, next coming to renal and urogenital embryology. Always remember, like I said previously, whenever you have any defects in development of the Mullerian or the Wolffian ducts, always go back and have a look at the development of the kidneys, because the development of these both is very closely associated. So the renal and urogenital system, they develop from the intermediate mesoderm. Now, development of the kidney takes place in 3 parts. So you have pronephros, mesonephros, and metanephros. Pronephros is the structure that appears initially, the first structure that appears initially. It is non-functional, it regresses completely. No filtration is happening, nothing is happening at that point of time. It regresses by week 3. Then mesonephros develops from week 4 to 8. It is functional for a very brief period of time. And this mesonephros, you have formation of the mesonephric ducts and tubules. Okay, so you have ducts and you have tubules. Tubules regress completely. Okay, they regress. In males, of course, the ducts are important, and the ducts are important in female also, because like I told you, they give rise to formation of the trigone of the bladder. So it is not that mesonephric ducts are completely unimportant in the female. In the female also, mesonephric ducts are needed because they give rise to formation of the trigone. Please imprint this on your brains. Now, this mesonephric duct, it gives rise to something what is called as the ureteric bud. This ureteric bud and the metanephric mesoderm that is there surrounding the mesonephric duct, it forms the metanephros, or what is called as the permanent kidney. Okay, so please remember, ureteric bud, it is a developmental structure from the mesonephros and not from the metanephros. Ureteric bud comes from mesonephros as an outgrowth. So metanephros is going to form the permanent kidney. So ureteric bud, it comes from the mesonephric or Wolffian duct. It gives rise to the collecting system basically in the kidney: ureter, pelvis, calycis, and the collecting ducts. And the surrounding metanephric blastema that is there gives rise to the rest of the kidney. So glomerulus, tubules up to the distal convoluted tubules. All this forms from the metanephric blastema. Now, if there is failure of formation of the ureteric bud, or this ureteric bud, it doesn't connect with the metanephric blastema, you will get what is called as renal agenesis. That means the kidneys fail to develop. The earliest time when filtration occurs is at about 10 weeks of gestation. The kidneys, when they start forming in the pelvis and they rise up progressively. So they start developing at about S1 to S2, but in the adult, the kidneys arise and then the final destination is L1 to L3. We know lower kidney is at a higher level. The right kidney, because of the liver, the ascent is obstructed and the right kidney is at a lower level than the left kidney. Now coming to something that is called as a horseshoe-shaped kidney. Okay, so this horseshoe shaped kidney, we know it is placed not in the normal position. It is not between L1 to L3. It is at a lower level. Why it is at a lower level? Because remember, uh, in the supposing this is the please excuse my drawing. My drawing is not good at all. It is just I'm for your understanding that I am drawing. Okay, so if you consider this is the developing spine of the fetus here, you will have your kidneys developing on either side. Okay, you have two separate kidneys developing. Now, because of some development anomaly that is happening, you have your spine here and you have your kidneys which are both conjoined. They are joined to each other. So you get formation of what is called as the horseshoe shaped kidney. Kidneys, we saw, they start developing in the pelvis and then they start rising. They go and for the final destination is L1 to L3. But what is happening, remember here, for a horseshoe shaped kidney, you have the IMA, that is the inferior mesenteric artery. Now, this inferior mesenteric artery, it is coming down. But your kidneys want to go up. But because they are joined like this inferior mesenteric artery, it prevents the kidney, this horseshoe-shaped kidney, from rising up. So that is why horseshoe-shaped kidneys are usually present in the pelvis. They are not present at the normal location of between L1 to L3. Something that you have to remember of the bladder trigone, that it develops from the mesonephric duct. Okay, so and the differentiation of the bladder trigone from the rest of the bladder is that the bladder trigone is smooth, whereas the rest of the bladder you will see formation of rugae. The rest of the bladder comes from the urogenital sinus, anterior part of the cloaca, the urogenital sinus, which is of endodermal origin. Important timelines to remember. The ureteric bud arises from the mesonephric or Wolffian ducts at 4 to 6 weeks. And then the cloaca, it gets divided by the urorectal septum at week 6 into urogenital sinus and anorectal canal. So urogenital sinus is anterior, anorectal canal is posterior. Now this urogenital sinus, it gives part to the lower urogenital system. So the urethra, lower 1/3 of vagina and the bladder, except of course the trigone. Remember, trigone comes from mesonephric duct. Now, if something goes wrong at this point of development, that means there is no connection in between the urogenital sinus and the mesonephric or the Mullerian ducts, you get ovira malformation. So what is this? You have an obstructed hemivagina and there is ipsilateral renal agenesis. This happens at around 10 weeks. So that is why I'm always saying whenever you find any development anomalies in the Mullerian system, please go back, have a look at the renal system. Always, most of the times, there will be some anomalous formation of the renal system also. Okay. Renal anomalies are usually seen in about 30 to 50% of Mullerian duct anomalies. We saw that there is something called as a renal agenesis. So the kidneys, they fail to develop. That is called as a Potter syndrome or Potter sequence. Kidneys are not developing. We know kidneys are one of the important sources of formation of amniotic fluid. So if the kidneys are not developed, you will not get any amniotic fluid. So that leads to oligohydramnios. Amniotic fluid is very important for development of the lungs. The fetus keeps on swallowing and there has to be plenty of amniotic fluid for proper lung formation to occur, and also it is important for formation of the limbs. So if there is oligohydramnios, you get pulmonary hypoplasia and limb defect. So this complete triad of bilateral region renal agenesis leading to oligohydramnios and pulmonary hypoplasia and limb defects, this is called as Potter sequence or Potter syndrome. Coming to the recall, important points. So you have the kidney developing from three structures: pronephros, mesonephros, and metanephros. All of these develop from the intermediate mesoderm. Mesonephros is something that is transient, partially functional. It gives rise to the duct and tubules. Tubules completely regress, and then you have the Wolffian duct, which forms a part of the male genital system, and also it gives rise to the bladder triangle. Metanephros forms the permanent kidney. Ureteric blood, please remember, it comes from the mesonephric duct. Okay, it does not arise from the metanephros, but this ureteric bud and the surrounding plastema, they form the adult kidney. Bladder trigone, it comes from the mesonephric duct. It does not come from the cloaca, or it does not come from the urogenital sinus. Horseshoe-shaped kidney is trapped under the inferior mesenteric artery at the level of L3. It doesn't ascend higher to L1. Ovira formation you get at 10 weeks, and Potter sequences because of bilateral renal oogenesis. Now coming to sexual differentiation and disorders of sexual differentiation. Previously, it was called as intersex. So to know this, you have to first know about how the sex is determined in the developing fetus. The key points is genetic sex is always determined at fertilization. So if depending on what chromosome comes from the sperm, if there are two X chromosomes, one X from the ovum, one from the sperm, then you get XX. If it is a Y chromosome, then you get XY. So it is determined at birth, the sex of the fetus. Then gonadal sex differentiation, that means whether it is going to lead to formation of the ovaries or the testis, it happens at 7 weeks. Now, how is that determined? So you have the SRY gene that is present on the Y chromosome. Okay, so if the Y chromosome is there, you will have this SRY gene, and this SRY gene, it gives rise to something what is called as the testis determining factor, or TDF. Now, if this is absent, if TDF is absent, then by default, because there is no SRY, you will get formation of the ovary. Okay. Now, once you have this SRY and you have the testis determining factor, you get formation of the testis. So in the testis, testosterone is produced by the Leydig cells. Now this testosterone, it induces, or it makes it, the Wolffian ducts are very sensitive to the presence of this testosterone, and they give rise to the male genital system. So the vas, seminal vesicle, and epididymis, all of these, they develop because of the influence of testosterone. Now the Sertoli cells that are there in the testis, they secrete what is called as anti-Mullerian hormone. Anti-Mullerian. So it acts against the Mullerian system, and there is regression of the Mullerian system. So female genital organs are not formed, and you get formation only of the male genital organs. Now, you have this enzyme which is called as 5-alpha reductase. This 5-alpha reductase, it acts on the testosterone. Testosterone, as we know, it is the inactive form. It has to be converted to dihydrotestosterone, which is the active form of testosterone. Under the influence of DHT, you get development of male external genitalia and development of the prostate. Okay. So remember that whenever there is no SRY gene or there is no Y chromosome, then by default you will get formation of the ovary. Because there is no testosterone, ovaries are not producing testosterone, there is no anti-Mullerian hormone, because no Sertoli cells are there. So that is why whenever there are ovaries, you get formation of the Mullerian duct or the female genital system, and then the Wolffian regresses. One more thing you have to implant in your mind is that Mullerian system, Mullerian, is also called as paramesonephric, or Wolffian is called as mesonephric. Okay, these two things you have to remember. This is the same thing in this diagram which I explained to you, and the gonads that are there at 5 weeks, they are bipotential. That means they could either develop into the testis or into the ovary. But if SRY is there on the Y chromosome, and that leads to formation of the testis determining factor, you get formation of testis and you get the male phenotype and development of the male genital system. If there is no SRY, then you get formation of the ovaries and the female genital system. Okay. Now coming to disorders of sexual development. So previously it was called as intersex, but now we call it as disorders of sexual development. The ones that you should be aware of is Turner, Klinefelter, and complete androgen insensitivity. Now, Turner is 45X, or it is also called as monosomy X, because remember, normally in our cells, we have, if it is a female, it should be 46XX. But when you have Turner's syndrome, it is 45X. That is, one X chromosome is lost, or whatever, Y chromosome. It is only one X chromosome that is there. So there is no Y chromosome. So it is a female phenotype. The ovaries that are developing, they do not develop normally. You have only streak ovaries. And there are a lot of other phenotypes that you see here. So the stature is short, there is neck webbing, there's coarctation of aorta, there are spider nevi, all these, and you have the chest, widely spaced nipples, cafe au lait spots, all these are present in Turner syndrome. Then Klinefelter, yes, it is male, but remember there is an extra X chromosome. It is 47XXY, because Y is there, one Y is there. So that is why genotype and phenotype, both of them, they are male. You have formation of the male external genitalia, everything is there. And this is sometimes they do present as infertility, and that is the time when they are diagnosed or discovered. They have normal mentation, it is compatible with normal life. These are male, unusually tall. They could be having gynecomastia. The testes are small. Remember, there is primary testicular failure in Klinefelter's syndrome. They present at about puberty, or later on they present as infertility, and you have very low testosterone levels in these individuals. Complete androgen insensitivity syndrome is XY, but the testosterone that is there, the receptors are not responding to testosterone. That is why you get androgen insensitivity. And because the testosterone receptors are not there, there is no development of the male external genitalia. External genitalia develop like a female, but there is formation of testes. Now, so we just saw all this about Turner. We saw that it is a female phenotype, streak ovaries, short stature, neck webbing, coarctation of the aorta. In Klinefelter's, it is 47XXY. A Y chromosome is present. So it is genotype, phenotype, both are males. These males are unusually tall. They have gynecomastia. There are small testes, infertile. And very important to remember, there is primary testicular failure. So testosterone levels are very low. And when testosterone levels are very low, remember, you will have high levels of LH and FSH, and also high levels of sex hormone binding globulins. That is another SPA in your endocrinology exam. So coming to complete androgen insensitivity. Okay. Complete androgen insensitivity, these are basically genetic males. Okay, they have a Y chromosome. Because a Y chromosome is there and you have the testis determining factor, that is why Mullerian ducts are completely regressed. Okay, but there is a defect in the androgen receptor. So you have a female genotype, that means male external genitalia do not develop, and because Mullerian is also lost, you don't have formation of a uterus, cervix, tubes, nothing is there. But testes are developing, and these testes, they do not develop in the normal place. There is no downward flow of the testes. They remain intra-abdominal. There is no descent of the testes into the scrotum because the scrotum is not forming. These testes, they remain intra-abdominal, and because there is a risk of them turning malignant. So once you have diagnosed, usually these complete androgen insensitivity, they present as primary amenorrhea. And once they are diagnosed, then after puberty, they are advised to undergo a gonadectomy because you want to decrease the risk of malignancy. Okay. Then congenital adrenal hyperplasia. Here also it is called as disorders of sexual development. In the male, nothing will happen because there are excess androgens. So you might get you will not get ambiguous genitalia, you will get normal formation of the genitalia. But if it is a female fetus and there is 21 hydroxylase deficiency, Okay, so what is happening in the adrenal is that there is increased production of androgens. There is no formation of glucocorticoids and mineralocorticoids. So whatever cholesterol is there, all that gets completely formed into androgens. And because of the excessive androgens, you get virilization, and ambiguous genitalia are there at birth in the female fetus. And later on, if they continue to thrive, you will get virilization. If they are not treated, it will give rise to virilization. Because there is no formation of mineralocorticoids and glucocorticoids, remember, someone with 21 hydroxylase deficiency, it is an emergency, it is a life-threatening condition. If not diagnosed immediately, it gives rise to salt wasting and severe hypoglycemia, and you might lose the baby. So it is very important that it is diagnosed and treated immediately at birth. Then 5-alpha reductase deficiency, it is another cause for complete androgen insensitivity. Okay, because we saw that testosterone has to be converted to dihydrotestosterone for it to be effective. Now, if there is deficiency of 5-alpha reductase, so at birth, even though it is an XY fetus, at birth, because testosterone is not affecting, external genitalia are not developing like that of a male. So you have a female at birth. At puberty, what happens? There is a surge in the testosterone levels. These are being produced in a very huge amount, and that is why you get masculinization at the time of puberty. And that is the time they are discovered or they are diagnosed as 5-alpha reductase deficiency. Okay. So important points to remember here is that whenever there is no SRY gene, you get formation of the ovary. Whenever you have SRY gene and testosterone, that gives to development of the Wolffian ducts. Dihydrotestosterone is necessary for development of external male genitalia, and under the influence of anti-Mullerian hormone, Mullerian duct regresses. So this is the same thing that we spoke about right now. Now coming to teratology and the critical periods, and what about torch infection? So the period of organogenesis, that means when all the major organ systems are being formed in the fetus, it is between week 3 to 8. Okay. Before 3 weeks, you have something that is called as all or nothing, all or none phenomenon. What is this all or nothing phenomenon? So before 3 weeks, what happens if there is some external injury? It could be in the form of drugs, radiation, or whatever external injury that is happening to the developing fetus. Before 2 weeks, the cells that are there, they are totipotent cells. That means they can develop into any system. So if a little bit of the cells are destroyed before 2 weeks, the rest of the cells, they can cover up for these cells. Okay. So if it is just an insult that is mild, nothing will happen. This pregnancy escapes and it goes on to form a normal pregnancy. But if the injury or the insult that is there, which is very severe, it's a very severe injury or an insult, then all the cells die and the pregnancy miscarries. So that is what is called as a all or none phenomenon. It happens before 2 weeks. From 3 to 8 weeks, if there is some injury that happens, then you will get some functional defects. Again, here, if it is a very severe injury, you can get the fetus can miscarry. But if there are minor injuries that happen during the period of organogenesis, then you get some functional defects. That means some defect in the growth or some defect in the behavior, but not major structural defects, because after week 8, okay, between 3 to 8 weeks, again, if you get some injury, it is going to be some major structural injury. By week 8, all the structures have been completely developed. So growth of the structures is going to be affected. Now, torch infections. So perinatal infections, as we call them, these infections, they can cross over from the placenta and they can cause fetal damage. Which are these organisms? So T is for toxoplasmosis, O is others, that means syphilis, varicella, parvovirus, HIV, Zika, R is rubella, C is CMV, cytomegalo, and H is herpes simplex. Now, what happens with infection with rubella? Remember, it has to be a primary infection. Secondary infections will not lead to this because mother already has formation of immunoglobulins. So, a primary infection, rubella infection, in this critical period of organogenesis, if you have in the fetus, you will get cataract, you will get cardiac defects in the form of PDA, deafness, and microcephaly. So, rubella and CMV, they are the most common causes of sensory neural hearing loss. But nowadays, because of a very good drive against rubella vaccinations, rubella has become less of a cause for sensory neural hearing loss, and it is CMV now, which is the most common cause for sensory neural hearing loss in the fetus. So CMV, you get hearing loss, you get periventricular calcification, and you will also get fetal growth restriction or IUGR. With toxoplasma in the fetus, there is hydrocephalus, chorioretinitis, intracranial calcifications, parvovirus or parvovirus B19. Remember, with parvovirus, there are no anomalies that develop in the fetus. This parvovirus, it basically attacks the RBCs. So it gives rise to severe anemia. It gives rise to severe fetal anemia. If this fetal anemia is not recognized, it is not treated, it can give rise to hydrops, non-immune hydrops in the fetus, ultimately leading to intrauterine fetal death. But if this hydrops is recognized early and you treat it with intrauterine transfusions and the fetus survives, you will not find any anomaly in the fetus. So it's very important to remember parvovirus only gives rise to fetal anemia. With Zika virus, you get microcephaly and brain malformations. Infection with varicella, you get fetal varicella syndrome. It happens usually if the mother is infected prior to 20 weeks of pregnancy. And then you get skin scarring, limb hypoplasia, eye defects, CNS defects. With syphilis, you can get stillbirth, you can get the characteristic teeth that are there, Hutchinson's teeth, you can get a sandal nose. So these are all the infective causes. Then there are a lot of drugs which are teratogenic. So warfarin, valproate, retinoids, methotrexate, ACE inhibitors, lithium, all these drugs are teratogenic. So that is why we usually avoid them in pregnancy. Remember, valproate, it has the worst teratogenic profile, and you will normally avoid valproate for any pregnant lady. So any woman with epilepsy, if she is on valproate, it is stopped in the preconceptional period, and she is given some other drug which is baby friendly. So valproate gives rise to neural tube defects, and also it can give rise to autism. So valproate is something that is always avoided in pregnancy. The takeaway message from this slide is that the period of organogenesis, that means week 3 to 8, that is the very critical period. Any insult that is there to the growing fetus or embryo at this point of time, it gives rise to major malformations. Prior to week 8, you have what is called as the all or none phenomenon. After 8 weeks, there will be only functional defects. But between week 3 to week 8, that is the most critical period, because at that time, you are having formation of all the major systems. So organogenesis critical period is week 3 to 8. Before week 3, you have all or none. CMV is the commonest congenital infection. With rubella, you can get cataract, PDA, and deafness. With toxo, there is hydrocephalus, chorioretinitis. With parvovirus, fetal anemia, and hydrops, no congenital anomalies are there. Zika, commonly giving rise to microcephaly, valproate, neural tube defects. That is why valproate always avoided in pregnancy. Warfarin, chondrodysplasia punctata, very common, especially given in the first trimester, 6 to 9 weeks. That is the period where warfarin is said to be most teratogenic. Lithium, you have Epstein's anomaly. That means there is a single ventricle that is functioning. All right. Now, let us see some of the most common SBAs that are there. from embryology. Fertilization normally occurs in which part of the fallopian tube? I think by now you should know by default fertilization occurs in the ampulla. Okay, it occurs in the ampulla and then implantation occurs in the uterus. So whenever if the if this developing zygote it's not transported from the ampulla to the uterus, ampulla is the commonest site where you get your ectopic. Adrenal medulla is derived from which embryological tissue? So medulla doesn't come from the mesonephros, it comes from neural crest. Please remember that adrenal medulla comes from the neural crest, adrenal cortex, it comes from the mesoderm. Please remember this, it is a very classic exam track. Bladder trigone is derived from where? Most part of the bladder, the other part of the bladder except the trigone is derived from the urogenital sinus. But the trigone, please remember, it comes from the mesonephric or the Wolffian duct. And it looks distinct from the rest of the bladder because it has a smooth surface at the level of the trigone, whereas rest of the bladder, it has the epithelium is thrown into folds or what we call as rugae. Monozygotic twin splitting at day 5 produces which chorionicity? So day 5, remember, already the inner cell mass and the outer cell mass has formed. So you will not get DCDA twins, but you will get MCDA. It means monochorionic diamniotic twins. Day 4 to 8 split, you will get MCDA. Then before that, day 0 to 3, DCDA. 8 to 13, you will get MCMA. After day 13, you will get conjoint twins. And whenever you have the T sign on ultrasound or you have a very thin membrane, it is a classic sign of MCDA twins. Foramen oval closes functionally at, remember foramen oval, it is present only in the fetus in utero because the right side of the heart is a very high pressure system and left side is a low pressure system. At birth, the reverse happens. Left side of the heart becomes high pressure, right side becomes low pressure. So foramen oval, functionally it closes at birth. At the first breath, foramen oval closes functionally. Anatomical closure occurs a little bit later, where ductus arteriosus closes at 24 to 72 hours after birth. It doesn't close at the first breath. Foramen oval closes at first breath. Overa syndrome arises from developmental defect at 10 weeks. You have to know this, okay? So what is overa? It is obstructed hemivagina and ipsilateral renal agenesis. The defect at 10 weeks because the ureteric bud, it fails to interact with the metanephros. We already saw that in development of the renal system. Karyotype of complete androgen insensitivity syndrome. Remember, these are males. So it will be 46XY. Okay. Androgen insensitivity is there because of the defective androgen receptors. Because androgens are present or the SRY is present, AMH is present. So that is why the testes form because of SRY. Because of AMH, Mullerian regresses, so no uterus, but you get formation of testes. There is no androgen response, so there is female external, the phenotype is that of a female, and you have testes which are intra-abdominal, and because they are in an ectopic place, there is a risk of malignancy developing, so you will remove them, or gonadectomy is done after puberty. Most common congenital infection causing fetal damage. Commonest is rubella and CMV, but nowadays it is CMV because of the massive drive that is taken for rubella vaccination. So it is always CMV, the most common cause of sensory neural hearing loss and also periventricular calcifications, fetal growth restriction. Critical period for major structural malformations during organogenesis. Remember, period of organogenesis is between 3 to 8 weeks. Before week 3, you will have all or none. That means if the pregnancy continues to grow, there will be no anomalies. If there is very severe injury, you will have a miscarriage. Okay. And after week 8, because all the organ systems have already been formed, you will have functional defects. You will not have any major malformations. Now, Mullerian or paramesonephric ducts. So, Mullerian or paramesonephric ducts give rise to all except Mullerian gives rise to the female structures. Okay. So fallopian tube, it's a female structure. It will arise from Mullerian. Uterus also arises from, it's a female structure, arises from the Mullerian. Cervix, also female, arises from Mullerian. Bladder trigone, okay, upper third of vagina. Even if you are not sure from where it comes, bladder trigone, what about bladder trigone? Where does it come from? It comes from mesonephric. I hope that is imbibed in your minds by now. So mesonephric. So the most correct answer here, if there is a confusion between these two, please remember bladder trigone comes from mesonephric. So the correct option here is bladder trigone. Okay. Upper 2/3 of vagina comes from the Mullerian, and the lower 1/3 it comes from the urogenital sinus. So that is all about embryology. I hope I was able to clear your doubts and I have made embryology a little bit easy for all of you. I'm just taking one break of 1 minute to get a glass of water. All right. So let's go ahead now, starting with genetics. In genetics, you have to know about cell division, you have to know about chromosomes. Very important, you have to know about the inheritance pattern. And you will be getting pedigree charts. So you have to know about how you are going to interpret the pedigree charts. That is the most common source of confusion for everybody in the exams. I will try to make it a little bit easy for all of you. So we will see cell division, cell cycle, cell division, Mendelian inheritance patterns, chromosomal abnormalities, non-Mendelian inheritance patterns, and then we will solve a few SPAs. So we know that cells are always dividing or replicating because that is how growth is going to take place. So you have to know about the phases that are there in cell cycle. It is G1, S, G2 and M. So girl says good morning. That is how you will remember the cell cycle. And remember, these three parts are called as the interphase. The cell spends most of the time in interphase. About 90% of the time it is interphase. Mitosis or meiosis, that is cell division, it's only about 10% of the time. So G1 is the cell is growing in size because it has to divide, right? And at that time, the organelles are also duplicating. Then S is for synthesis because when a cell is dividing, the DNA also has to divide. And we have mitosis and we have meiosis. So mitosis, two identical cells are being formed. So that is why during the synthesis period, DNA has to be replicated so that both the cells, both the daughter cells, they get equal amount of DNA from the parent cell. Then you have G2 or the error check for protein synthesis, and then you have mitosis. So for mitosis, there are four phases. It is the mnemonic that is there is PMAT, prophase, metaphase, anaphase, and telophase. The checkpoints that are there, now it is very important that you have checkpoints before the cell divides. Otherwise, what happens is you will have cells which are abnormal and they keep on dividing, and then that is how you get formation of cancers, or you will get formation of uncontrolled of cells that are not normal. So the checkpoints are there between G1 and S. Then you get another. So at any point of time, whenever you have at the checkpoint, if the cell realizes that whatever has happened in G1 or in the synthesis or something has gone wrong in mitosis, the cell stops dividing. It doesn't go forward into mitosis. So that is a natural defense mechanism by the body. I have given you a slide after this here, which shows what exactly is happening at the checkpoints. Okay. Then coming to the two types of cell division that we have, mitosis and meiosis. Mitosis, remember, it happens in all the somatic cells and it produces two identical diploid daughter cells. And here you have prophase, metaphase, anaphase, and telophase. How to remember? Prophase is the phase that is happening, that is the prophase or the beginning. Okay, so here the cell is preparing itself for division. Then metaphase is, I remember it as meet. So here what happens is all the chromosomes, they meet in the center of the cell. Anaphase, the chromosomes, they go away from each other. This is away, A for away. And telophase means you get formation of two cells or two nuclei. Once the two nuclei are formed, then you get cytokinesis and you get formation of two cells. So that is how you remember prophase, metaphase, anaphase, and telophase. Mitosis is basically used for growth and for tissue repairs. It's happening in all the somatic cells. Meiosis is something that happens in the gametes. It is called as the daughter cells that are produced after meiosis. They are not identical to the parent cell. The genetic material that is there in the daughter cells or after meiotic division, the genetic material is different from the parents. Okay. Meiosis is also called as reductional division or unequal division. There are two parts in meiosis, meiosis 1 and meiosis 2. Meiosis 1 is the reductional division. Here also, even here you get PMAT again. Okay, you will get PMAT. Even in meiosis 2, you will get PMAT. So how do you differentiate? It is called prophase 1. When it is happening meiosis 1, you call it as prophase 1, metaphase 1, anaphase 1, or telophase 1. And when it is happening the second time, it is called prophase 2, metaphase 2, anaphase 2, and telophase 2. Okay. So meiosis 1 is the reductional division. What happens here in meiosis? In mitosis, remember what happens is the two sister chromatids, they are pulled apart to different parts of the cell. But in meiosis, homologous pairs of chromosomes, they align at the center of the cell and a complete chromosome is pulled to one side of the cell. So that is why you get cells that are formed, they have 23 chromosomes after meiosis 1. That is why it is called as the reductional division. Meiosis 2 again is a equational division. It is very similar to mitosis. And here, like in mitosis, there is separation of the chromatid. Okay. Crossing over or chiasma, it happens in prophase 1. So when two homologous chromosomes, they are aligned next to each other, there is some exchange of chromosomal material in between these two. So a part of this chromosome will go and attach here, and a part of this chromosome will come and attach here. So that is why the genetic material that is there in cells that are formed after meiosis, it is different from the genetic material of the parent cell. And this phenomenon, this crossing over or this chiasma that is produced, it explains why siblings are not identical and why they are different from each other. Because of the difference in the genetic material that is there after the homologous chromosomes, they align in meiosis 1. Now, if something goes wrong in meiosis 1, that means the chromosomes, there is non-disjunction happening. The chromosomes are not being separated. Then one gamete will have an extra chromosome. That is how you get formation of trisomies. So down, inward, and patau. Because there is non-disjunction in meiosis 1. It is commonly seen in meiosis 1. Very rarely it is seen in meiosis 2. Now, another important thing that you have to remember about this is that in the fetus, okay, whenever oogenesis, it starts in the utero, but it is not complete at that time. it is arrested in prophase 1, and this meiosis, it is complete at ovulation, and you get formation of the mature. Once fertilization is complete, then you get, once fertilization occurs, then you get formation of the second polar body. Please remember this. So the ovum, they are arrested at prophase 1. Once ovulation occurs, meiosis 1 is completed. Meiosis 2 is completed at fertilization. So this is the cell division or the checkpoints that are there. So you have checkpoint at G1, which is called as the restriction checkpoint. At this point, the cell sees whether the cell, because before going from G1 to S or synthesis, the cell has to know whether it is big enough for division or not, whether there is energy resources are enough or not, whether the damage, whether the DNA, is it normal or is it damaged? Now, if there is anything abnormal that is happening here, the cell doesn't continue into the further cycle. It goes back to G0 and it tries to repair the damage that is there. Then the other checkpoint that is there at G2. So at G2, the cell checks whether all the chromosomes have replicated correctly or not. It also checks again for DNA damage. If there is any damage here at any point of time, if the cell realizes that there is damage to the genetic material or there is damage to the DNA, further division does not happen. Then in the mitotic phase, at metaphase, again there is a checkpoint before formation of the two daughter cells. So at that point, the cell checks whether all the chromosomes, they are lined up correctly in the center of the cells and they are ready to be pulled apart. So at any phase of this between G1 and S, between G2 and metaphase or G2 and M, if the cell finds that something is going wrong, the cell doesn't go ahead with mitosis. Remember the 4 phases: prophase, metaphase, anaphase, and telophase. So to sum up, the cell cycle is G1S, G2M. So girl says good morning. S phase is for replication of the DNA. P53 is the guardian of the genome. Sometimes they might ask you that in an SBA. Mitosis produces 2 diploid identical cells. Meiosis, you get 4 haploid gametes, which are, the genetic material is completely different from the parent cell after meiosis. Meiosis 1 is reductional division, where the homologous chromosomes, they are pulled apart. So at the end of after meiosis, you get N is equal to 23 in all the cells. Meiosis 2 is equational, but again after meiosis 2 also, that is why gametes that are formed, they are haploid. Because why they are haploid? Because the ovum is 23, sperm is 23. So once fertilization again occurs, then you get formation of the diploid structure is again restored. So that is why gametes are haploid and they are not diploid. Crossing over happens in prophase 1 during meiosis. Whenever there is non-disjunction, it gives rise to trisomies. It is very common in meiosis 1 rather than in meiosis 2. And oocytes, they are arrested in prophase 1 until ovulation. After ovulation, prophase 1 is completed or meiosis 1 gets completed. It enters meiosis 2. Meiosis 2 is completed after fertilization. All right. So that is all about cell growth and division. Now coming to something which is very like most of the students find a difficulty in recognizing how to read the pedigree charts. Okay, so these charts are pedigree charts for the Mendelian inheritance patterns, that is autosomal dominant or autosomal recessive, X-linked dominant, X-linked recessive. For all these, it's very useful to read the pedigree chart. We will see how to read the pedigree chart. First, let us see what are the important characteristics that are there for autosomal dominant. Okay, so dominant means even one copy of an abnormal allele, it will be expressed because it is dominant, and that individual will show the disease pattern. Now, if one parent is affected, 50% of the offspring is going to be affected. Because it is autosomal, both sexes are equally affected, and there is vertical transmission. That means there is no escape, there is no skipping of generation. See, in each and every generation, you will see the disease. Because of variable penetration and expressivity, like not all individuals who are affected, they will show the complete range. There will be a variance in how they express the disease. Very common examples of autosomal dominant. They are Marfan's, Huntington, neurofibromatosis, achondroplasia, BRCA1, BRCA2, both are autosomal dominant. And then you have von Willebrand's and Lynch syndrome. Autosomal recessive. Recessive means they are expressed only in the homozygous form. That means both of the alleles that are there, they have to be abnormal so that the gene gets expressed. Now, when both parents are carriers, about the 25% of the offspring, that means 1 in 4 will show the disease pattern. Because it is autosomal recessive, both the sexes are equally affected, and autosomal recessive patterns are usually expressed in families that have consanguinity. So examples of this are cystic fibrosis, sickle cell disease, beta cell thalassemia, Tay-Sachs disease, PKU, phenylketonuria, congenital adrenal hyperplasia, Wilson's, and Friedreich's ataxia. These are the common diseases that are there with autosomal recessive. So remember autosomal recessive because a person needs to have both abnormal alleles. So the frequency of autosomal recessive that you see in a population is very less. Now coming to how you are going to read the pedigree charts. Okay, so you have to be aware of the symbols that are used in pedigree charts. If you know the symbols, then it is very easy of how you are going to read the pedigree charts. Just 1 minute. All right. So the symbols. Square is male. And if the square is not darkened, it is an unaffected male. Females are depicted as round as always. If it is not shaded, it is unaffected female. Square in a solid pattern is affected male. A circle, which is a solid, it is an affected female. If you have a circle with a dot in between, sometimes it is a carrier. Because it's a circle, it is a female. If you have a square with a dot in between, then it is a carrier male. Now, when you have these horizontal lines between male and female, it indicates that they are mating. And when you have the vertical lines, it shows that they are the offspring of this couple. So let us see autosomal dominant. Now remember, it is autosomal dominant. It is going to be expressed in each generation. There is no skipping of generations. Okay. And also a male can pass or transmit the disease to the male, female can transmit to female. And it is seen in 50%, at least 50% of the offspring will be affected. Okay, so now what we are seeing here is you have this affected male mating with a female that is unaffected. Okay, so this is the first generation. In second generation, what you are seeing, you have an affected male, you have an affected female, and you have an unaffected female. If you come down to the third generation, what you are seeing is this affected male, he is transmitting the disease again to an affected male. So whenever male to male transmission is there, or you are not seeing any skipping of generations. Whenever no skipping is seen, it is always a dominant trait. And whenever it is male to male, if you see, be rest assured that it is autosomal dominant. Okay. Now, autosomal recessive. Because it is recessive, you can see skipping of the generation. And for the gene to get expressed, both the parents have to be carriers. And if one of the offspring gets both the abnormal alleles, then you will get expression of the disease in that offspring. So now you have this affected male, you have this affected female. 20%, 25%, you will get one unaffected child. You will get one child that is an affected female. You will get one affected male. These are carriers. Carrier female, carrier male, and you will get one child that is affected. So they have shown here that this is an affected female. It could be an affected male also because it is autosomal. Okay, there is no question of X-linked or Y-linked. It is autosomal. So males and females are equally affected. And remember that the person has to have both the abnormal alleles to show the disease. So 25% of the offspring is affected when it is autosomal recessive, 25% is unaffected, and 25% is carriers. And it will usually be consanguineous. So whenever you see this double line, it indicates that it is a consanguineous marriage or consanguineous mating. Now coming to X-linked recessive. In X-linked recessive, remember, though it is X-linked, most of the times it is the male that is going to exhibit the signs and symptoms of the disease, and the females are usually carriers. Why? Because females, we know, they have two X chromosomes, right? There are two X chromosomes in a female. So even if one X chromosome is the affected chromosome, the female has another X chromosome which is normal and it is recessive. So it is going to be expressed only when both chromosomes are abnormal. So this is something phenomenon which happens very rarely. On the other hand, males have only one X chromosome and they have this X chromosome which is damaged or which is abnormal, and the other chromosome is the Y chromosome. Because they are having only one X chromosome, that X chromosome is carrying the damaged allele, they express the signs and symptoms of the disease. So let us see when you have a carrier mother. So she is having one affected X chromosome, other X chromosome is normal. She is not affected. She doesn't have the disease. And you have the male is completely normal. So this affected male, now what happens in the male? The male is going to get affected X chromosome from the mother. Y chromosome is going to come from the father. Y chromosome is normal. So this so here what happens, out of two males, one male is affected and one male is normal, because the female has one X chromosome which is affected, other X chromosome is normal. So one male gets the affected X chromosome, the other male gets the normal X chromosome. So one of the male is affected, other male is normal. You get one female which is carrier because she is going to get one affected X chromosome from the mother. The other X chromosome is coming from the father, which is normal. So because she is a carrier, she is not going to express the disease. And this female, she is going to get the other normal chromosome from the mother and another normal chromosome from the father. So in X-linked recessive, remember, there is no transmission from male to male, unlike autosomal dominant where you get transmission from male to male. In X-linked recessive, the transmission is always from affected female or a carrier female to a male. And this male, what is going to happen is the daughters of this affected male, they are going to be carriers. They are, unless this affected male again marries a female that is affected or marries a female that has the disease. If the partner of this affected male is a normal female, the carriers, the daughters are all going to be carriers and all the sons are going to be normal. Okay. Now coming to X-linked dominant. Remember, it is X-linked and it is dominant. So one X chromosome, one defective X chromosome is enough for the disease to be affected, to be expressed. So here you have the affected father and you have a normal mother. Okay. Now what is happening? The females, all the females are going to be affected. Because females, they are going to get one affected X chromosome from the father, and the normal X chromosome is going to get from the mother. But because it is a dominant trait, even one gene, one affected chromosome is enough for expression of the disease. Whereas the males, they are going to get only the Y chromosome from the father. So all the males are going to be normal, whereas all females are going to be affected. So whenever you get in the pedigree chart, father is affected and all the daughters are affected and all the males are escaping. They are not getting the disease. Then it is X-linked dominant. Okay. So I hope I try to make it as simple as possible for you. If you still have any doubts, you can tag me in the group and I would try to make it simpler for you. So let us see who gets what, how to recognize it on a pedigree chart. First, let us see X-linked recessive. So X-linked recessive, we saw males are more affected and the females are usually carriers. There is no male-to-male transmission. That is the key clue for them, no male-to-male transmission. All daughters of affected father, they are obligate carriers. Hemophilia A or B, muscular dystrophies, color blindness, fragile X, all these are examples of X-linked recessive. X-linked dominant is something which is more rarer. Both the sexes can be affected. Females are mildly affected because they have another normal X chromosome. Even an X-linked dominant, if you have an affected father, all the females, all the daughters are going to be affected and the sons are going to escape because they will get a normal X chromosome from mother, the Y chromosome comes from the father. So X-linked dominant, these are diseases which we see quite rarely in the community. Rett syndrome, vitamin D resistant rickets, Alport's, some of Alport's syndrome. Alport syndrome, the inheritance patterns are variable, and incontinentia pigmenti. All these are types of X-linked dominant. Now, Y-linked, Y chromosome is going to be transmitted only to the males. So whenever, again, you see transmission only from male, from the father to all sons, again, it will be Y-linked. And very few of them are there. So whenever you have micro chromosomes, micro deletions in the Y chromosome, leading to infertility, those are Y-linked disorders. Then mitochondrial inheritance. So one thing that you have to remember about the mitochondria is that when fertilization is taking place, all the mitochondria from the sperm, they are destroyed. And whatever mitochondria we inherit only from the maternal side. So if there is any mitochondrial disturbance in the male, that is not transmitted to the offspring at all, because all mitochondria from the male at the time of fertilization, they are completely destroyed. So mitochondrial disturbances, they are only maternal inheritance. All offspring of the mother, affected mother, she has some mitochondrial disturbance. All her offspring are going to be affected. There is no paternal transmission for this. And these are something which are very rarely seen, mitochondrial. So you get some optic neuropathies, then you get MELAS, or you get Kearns-Sayre syndrome. These are basically very rare syndromes that are seen. And they are seen whenever high energy organs, that means the brain, muscle, or the eyes, some disturbances in these systems, these are usually of mitochondrial inheritance. So let us again quickly go through the inheritance patterns. Autosomal dominant, remember, every generation is going to be affected. There is no skipping of generation because it is autosomal, both sexes are affected. There is male-to-male transmission. 50% of offspring, they get affected if one affected parent is there. If both are affected, then 75% of the offspring is affected. You get with autosomal dominant, Marfan's, Huntington, Neuro, neurofibromatosis, achondroplasia, BRCA1-2, Lynch, von Willebrand's, and familial hypercholesterolemia. Then autosomal recessive. So there is skipping of generation seen. Both sexes are equally affected because it is autosomal. Consanguinity is commonly seen in autosomal recessive disorders. And if both the parents are carriers, then 25% of the offspring, or 1 in 4, is affected. So cystic fibrosis, sickle cell beta thalassemia, Tay-Sachs disease, phenylketonuria, congenital adrenal hyperplasia, Wilson's, Friedreich's ataxia, galactosemia, Gaucher's, all of these are autosomal recessive. Now coming to X-linked recessive. So males are more affected than females. In the pedigree chart, when you see more of female affected rather than females are only carriers, then you know that it is an X-linked recessive pedigree. There is no male-to-male transfusion, something which is you have to remember. In X-linked recessive, there is no male-to-male transfusion. The carrier mother, she will transmit it to the affected male. Whenever you have an affected dad, all the daughters are carriers. And whenever you have a carrier mother, 50% of the sons get affected. 1 out of 2 of the son is affected. 1 of 2 of the male is normal. Then hemophilia, both types A and B, muscular dystrophy, speedy deficiency, color blindness, fragile legs, Bruton's. All these are types of X-linked recessive inheritance patterns. Mitochondrial inheritance. Because mitochondria from the male are all destroyed, mitochondrial inheritance is maternal transmission only. If the dad is affected, none of the children will be affected. If the mom is affected, all the children, all offspring are at risk. And you get something that is called as heteroplasmy. That means the severity is different among individuals also. Not all of them will have the same severity of the disease. The expression is variable. Mitochondrial is something that you very rarely see. So you have the liver hereditary optic neuropathy called as Lehon, or you have Miller's, Murph, Kearns side. Important thing is wherever any organs that require very high energy, brain or the eyes or muscles, so all of these systems could be affected whenever you have the mitochondrial disturbances. So this is all about the mitochondrial disturbance. I've already explained to you why the transmission is only through the maternal. Now, as humans, we are really very highly independent, highly intelligent. We have found out a way how you are going to escape this mitochondrial diseases. So whenever you are doing IVF, you will do 3-parent IVF. You have a male or a female in which there are mitochondrial disturbances, not a male, because if there is in the male any disturbance in the mitochondria in the male, it's not going to be transmitted to the offspring. But in the female, then only that mitochondria, the defective mitochondrial gene, that is replaced, and you get something that is called as 3-parent IVF, what is called as mitochondrial replacement therapy. So briefly to recall the inheritance patterns, Mendelian inheritance patterns. Whenever you have autosomal dominant, 50% of the offspring are affected. The transmission is vertical. Both sexes are affected. That means there is no skipping of generation. In autosomal recessive, 25% of the offspring are affected. It's usually seen in consanguineous marriages. X-linked recessive, males are affected. There is no male-to-male transmission. In X-linked dominant, if the father is affected, all the daughters are going to be affected, the sons are going to escape. If it is Y-linked, the transmission is only from father to son. Mitochondrial inheritance pattern is only from the mother. Okay. Common autosomal dominant trait or diseases are Marfan's, Huntington's, neurofibromatosis type 1, BRCA, and Lynch syndrome. Common autosomal recessive is cystic fibrosis, sickle cell beta thalassemia, Tay-Sachs. Common X-linked recessive is hemophilia A and B, muscular dystrophies, G6PD, and Fragile X. Common X-linked is Rett syndrome and vitamin D resistant rickets. All right. Now, after we have seen Mendelian inheritance patterns, let us see some chromosomal abnormalities. So, whenever there is abnormality in the number of chromosomes, we call it as aneuploidy. So, there is abnormal chromosome number. Trisomies are the ones which are more common. Trisomy 21, or what we call as Down. The incidence in the population is about 1 in 700 births. There are typical characteristics seen in the newborn. There is a flat face, epicanthal folds are there. There is a single palmar crease, atrioventricular septal defects, and duodenal atresia. Now, the risk of trisomy 1, it increases as the maternal age increases because the ovum is getting aged, and we saw that if there are defects in meiosis 1, then it can give rise to Down's syndrome. So it can give rise to trisomies. And among the trisomies, the commonest one is trisomy 21, or what we call as Down syndrome. Now, other than defects in like disjunction at meiosis, there is another method in which you can get Down syndrome. If there is a translocation happening, or Robertsonian translocation, which happens in acrocentric chromosomes. So 13, 14, 15, 21, 22. These acrocentric chromosomes, if translocation happens in these chromosomes, and if one of the parent is a carrier of this Robertsonian transmission, this Robertsonian translocation, then you could be getting repeated episodes of Down syndrome in the offspring. Otherwise, usually we see that Down syndrome, there is no risk. The risk of recurrence is something which is very rare. But if you have a paternal cause, like either the mother or the father is a carrier of Robertsonian translocation, then the risk of recurrence of Down syndrome is high. Then you have trisomy 18. That means there are 3 chromosomes. Chromosome number 18, you have 3 of them. It is called as Edwards. It is more rare than Downs, 1 in 6,000 births. It has again very characteristic features. There are clenched fists. That means at ultrasound, you will see the fists. Normally in the fetus, the fists are open. If you see clenched fists at ultrasound, at the anomaly scan, you have rocker bottom feet. There are cardiac defects, severe IUGR seen at anomaly scan. Normally, IUGR develops after 28 weeks of pregnancy. But if you are seeing IUGR developing early, and you have all these other things that are seen, rocker feet or clenched fist, some cardiac anomalies, it should ring a bell, and it could be trisomy 18. Most of them usually do not see the first birthday. Then trisomy 13, or what we call as Patau, is something which is still more rarer. It is about 1 in 10,000. Most of the features are similar to Edwards. You will get severe IUGR, but midline defects are more common with trisomies. So cleft lip or palate, defects in the development of the brain, holoprosencephaly, polydactyly, these are more common with trisomy. Then another type of chromosomal abnormalities, we've already seen about Turner, and we have already seen about Klinefelter. So I will not repeat the same thing again and again. Another thing that you have to remember for Turner is that they could be mosaics. So they could be having 2 cell lines. One cell line is 45X. The other cell line is normal, 46XX. That is called as mosaicism. They have 2 types of cell lines. For Turners also, it is something which we see very rarely, 1 in 2,500, because most of the conceptors, 99% of them, they are lost early. So that is why at birth, it is only 1 in 2,000. And Klinefelters, it is about 1 in 600 males. The other thing that you have to remember about Klinefelters is that the risk of CA breast, there is gynecomastia that is seen in Klinefelters. So they have a high risk of CA breast as compared to the other normal males. And these males who have Klinefelter syndrome, because they have low levels of testosterone, because it is primary testicular atrophy, they benefit from testosterone replacement therapy. So this is again the same thing that we saw about chromosomal abnormalities. A few other chromosomal abnormalities that you have to be aware of is triple X 47. There are, it's an X chromosome, or these are sometimes called as super females. They are usually phenotypically they are normal females because there is no X chromosome. And then you have 46 XYY, another extra Y chromosome. It is called as Jacob syndrome. These are again tall, but unlike Klinefelter, where you do not get intellectual disabilities in Klinefelters. They have normal mentation, they have normal life. But those with Jacob, or where there is an extra Y chromosome, they could be having low IQs and they could be having some learning difficulties that are there in Jacobs, which are not there in Klinefelters. In Klinefelters, they have normal mentation. Then next, after numerical, you have structural anomalies in the chromosomes. So whenever there is deletion in chromosome 22, Q11, it gives rise to what is called as DGeorge. So you have cardiac anomalies, anomalies of the thymus or parathyroid anomalies. Whenever there is depletion of 5p, it gives rise to Cre-Du-Chat. Whenever there is translocation, so chromosome T, whenever in chromosome 9 and 22, whenever there is interchangeable, and you get formation of what is called as Philadelphia chromosome. So these individuals are at a very high risk of developing CML. And the thing to remember about Robertsonian translocation, that it involves acrocentric chromosomes. So 13, 14, 15, 21, and 22. So the parents, those who have this translocation, especially if it is a balanced translocation, the parents are normal. But if they pass it on to the offspring, that is the time when you can get downs. Most commonly, it is down. Very rarely, you will get trisomy 13 or patau. 18, trisomy 18 is Edward and trisomy 13 is patau. So Robertsonian translocation can very rarely give rise to trisomy 13. Now coming to a quick recall. So trisomy 21 or Downs. The risk is 1 in 700 live births, atrioventricular septal defects, duodenal atresia. Remember the double bubble sign that you see on ultrasound or even when an X-ray abdomen is done. T18 or trisomy 18 is Edwards, so clenched hands, rocker bottom feet, and with fetal growth restriction. T13 is Patau, holoprosencephaly, cleft lip and palate, polydactyly, again with severe fetal growth, early fetal growth restriction. That is either Patau or Edwards. Turner 45X, it is also called as monosomy X. So you get short stature, coarctation of the aorta, streak ovaries. Klinefelters, extra, you could say extra X chromosome because they are phenotypically males and also genotypically because a male Y chromosome is there. So they are tall, infertile, and primary testicular failure leading to low testosterone levels. Whenever there is 22q11 deletion, you get DGeorge. 5p deletion gives rise to Cre-Du-Chat. Philadelphia chromosome is whenever there is translocation between chromosomes 9 and 22. Robertsonian translocation occurs in between acrocentric chromosomes 13, 14, 15, 21, and 22. And whenever you have Robertsonian translocation in either of the mother or the father, so the parents are carriers of this translocation, there is a very high risk of having repeated Downs in the offspring. Now coming to non-Mendelian inheritance patterns. So here you have mitochondrial inheritance for genomic imprinting and anticipation. So mitochondrial inheritance, we already saw that only it's transmitted from the mother because the mitochondria in the sperm, all of them are destroyed. All offspring of the affected mother, they are at risk because the sperm contribute nothing, no contribution from the sperm to the mitochondria. Then there is heteroplasmy or there is variable severity. That means the offspring, not all the offspring will show the same affection pattern. There could be a variability. Examples we already saw, Lehans, Millas, Murph. Now, genomic imprinting. So, the classic example of this is Prader-Willi or Angelman. What happens in genomic imprinting is that expression of the gene, it depends on the parent of origin. Like either the defect is there in the mother or in the father. Okay. So, in Prader-Willi, what happens is there is deletion of the same the defect is the same chromosomal region 15q11 to 13. That is the region that is affected, but the phenotype that is there, it depends on which parent is affected. So in Prader-Willi, P for papa, the paternal 15q11 deletion occurs, or there is maternal uniparental trisomy. Paternal genes are not expressed in Prader-Willi. So Prader, P for Papa. So the paternal genes are destroyed. In Angelman, M for Mama. So here the paternal genes are present, maternal are destroyed. Prader-Willi, paternal is destroyed. In Angelman, maternal is destroyed. Now, what are the features? In Prader-Willi, you get a floppy infant, or because there is severe hypotonia, they do not thrive in infancy, but as they grow up, they develop hyperphagia, so they develop obesity. The gonads are very small. There is hypogonadism leading to infertility. They have almond-shaped eyes, small hands and feet. Even the intellectual disability is there, though not very severe mental retardation. It is mild to moderate severe intellectual disability. There is short stature and obsessive behavior. So the important thing to remember here is in Prader-Willi, there is paternal deletion. Okay. The father, the genes that are coming from the father, these are the ones that are destroyed, or there is no expression of paternal genes. Whereas in Angelman, the genes from the mother, they are destroyed, and the genes from the father, they get expressed. Angelman, it is also called as happy puppet because these individuals, they have inappropriate laughter, they are smiling all the time. There is very severe intellectual disability, ataxia, jerky movements, epilepsy, even seizures are there in 80% of the individuals. Speech is limited. They are sometimes, they cannot speak, or sometimes very severely affected speech is there. Microcephaly and prognathy, all these are there. So remember, the same chromosomal region is affected, but depending on whether there is loss of maternal or paternal genes, the syndrome is different. Now, anticipation. So what happens is a classic example is given of that of Huntington's, or in anticipation, what happens that the defect is there in the parents, but in successive generations, the severity of the disease goes on increasing. That is called as anticipation. Okay, because what happens in anticipation, there is repeated expression of the trinucleotides, and that goes on increasing in successive generations. The classic case is given that of Huntington's, where there is repetition of CAG, and successive generations you will have earlier onset, and mainly it is paternal expansion. And this Huntington's, as we already saw, is autosomal dominant. So in non-Mendelian or in anticipation, what happens? The disease comes on earlier in successive generations, or it is more severe in successive generations. It is due to trinucleotide repeat expansion. Classic case is there of Huntington's, in which the CAG is the trinucleotide that gets repeatedly expressed. Earlier onset and mainly paternal expansion for Huntington's. Myotonic dystrophy, the trinucleotide that is repeated is CTG. Here it is mainly maternal. For Fragile X, the trinucleotide that gets repeated is CGG. The premutation is there in the mother, but full expression you will see in the offspring. So that is why successive offspring or successive like from the parent to the children and then from the children again. So the third or fourth generation, you will see more severe disease. Even Friedreich's ataxia, that is also a type of anticipation in which the trinucleotide that is repeated is GAA. Now, uniparental disomy or UPD, we saw already Prader-Willi and Angelman. Then you have germline mosaicism. So that means it is the gonadal cells, the cells that are there in the gonads, they are affected and somatic cells are normal. So some cells in the gonad are affected, some cells are normal. So that is why you get sporadic autosomal dominant diseases which are recurring in the same family. Not all the offspring are going to be affected. It is only sometimes it will be affected. And autosomal dominant, normally there is no skipping of generations. But if it is mosaics, then you can get skipping of generation. Now, epigenetics means there is some modification in the proteins, or there is some modification in the methylation or the histones. So these are heritable changes without a change in the DNA sequence. So this is all about non-Mendelian inheritance patterns. So to recall about a non-Mendelian inheritance, mitochondrial, the inheritance is maternal only. In Prader-Willi, the deletion is paternal 15q11. In Angelman, it is maternal 15q11 deletion. In Prader-Willi, you get hyperphagia plus hypogonadism. In Angelman, it is happy puppet plus seizures. Anticipation means there is trinucleotide expansion happening. Huntington's, it is CAG repeats. Myotonic dystrophy, it is CTG repeats. Fragile X, CGG repeats. And germline mosaicism explains some sporadic autosomal dominant. Let us see a few SBAs. Okay. So BRCA, what is the inheritance pattern? By now, I think it should be firmly imbibed that BRCA is autosomal dominant. Okay. So autosomal dominant in BRCA, the first degree relatives are at a 50% risk, but you need to have the first hit and the second hit. Okay, so that is why whenever you have the BRCA mutation, we know people, those who have the BRCA mutation, they are at a high risk of ovarian and breast cancers. Now, the Knudson's hypothesis that is there, that the first hit and the second hit. So whenever you have any faulty alleles that are there, you for the complete expression of these faulty alleles or formation of cancer syndromes, both the alleles, they have to be affected. Now, whenever any person has a faulty defective gene, BRCA1 or BRCA2, whatever it is, at birth, they have inherited one of the faulty allele. So the first hit is already there at birth. Okay. And because the first hit is already, the second hit happens very early in life. And that is why with BRCA, the onset of cancers or in any of the cancer syndromes, BRCA, Lynch, Peutz-Jeghers, whatever, Lee from any, the first hit is already inherited and the second hit occurs early. So in cancer syndromes, all the cancers that are seen, they appear early on in life. Normally ovarian and breast cancers, we say these are cancers of late age. But with BRCA1, BRCA2, or even with Lynch syndrome, endometrial cancers that happen, they happen earlier on, say around 35 to 40 years of age. That is because of this Knudsen hypothesis, which says that for the alleles, one of the affected alleles or the first hit is already inherited at birth, and the second hit, it happens early on in life. So that is why these cancers, they appear early on in life rather than late. Commonest gynecological malignancy in Lynch syndrome, it is endometrial. Okay, 40 to 60% lifetime risk is there. For ovarian, it is only about 10 to 12. Please remember, with BRCA, high risk of ovarian and breast, and with Lynch syndrome, it is endometrial cancer. Now, which are the mismatch repair genes that are implicated in Lynch syndrome? Please remember, it is MLH1, MSH2, MHS6, and PMS2. This could come as an SBA, which are the MMR genes that are implicated in Lynch syndrome. So you have to remember these mismatch repair genes. Which feature is most suggestive of X-linked recessive inheritance? Remember, X-linked recessive. Always affected mother and males are the one that are affected. Carrier mother and males are affected. Vertical transmission across generations, it is recessive. So you will not get vertical transmission. Consanguinity in parents, it could be there. Even in autosomal recessive, no male to male transmission. Yes. Affected father, all affected daughters. No. If it is dominant, then all daughters, and only affected females. Even that is not correct. So whenever you have no male to male transmission, it is an example of X-linked recessive. So father is affected. He will not transmit it to any of the males. The daughters will be carriers. Okay. Now you have an affected father with X-linked dominant disorder. Remember, X-linked dominant. What is the risk to his daughters? Understand, X-linked dominant, the daughters get one X chromosome from the mother, one X chromosome from the father. If the X chromosome in the father is affected and it is a dominant trait, so they are getting the defective X chromosome from the father. So 100%, okay, all the daughters will be affected because it is dominant. Okay, X-linked dominant, all the daughters are affected. And no son is affected because the sons are going to get the Y chromosome, which is normal, and the X chromosome from the mother. Now coming to trisomy 21 or Down syndrome screening, what happens to the combined test here? I think all of you know this, that in Downs you get too high and too low. So whenever in any of the biochemical markers, if H is present and if that is high, so too high and too low, HCG high, inhibin high, that is very characteristic of trisomy. And of course, you have to look at the nuchal translucency. So wherever the nuchal translucency is increased, there is increased risk of trisomy. So wherever the nuchal translucency is normal or it is decreased, it is definitely not Downs. So C, D, and E are out of question. A and B. Now in A, nuchal translucency is increased and HCG is also raised. Papa A is decreased. In B, raised nuchal translucency but low levels of HCG. HCG will be raised in trisomy 21. Okay. And also Papa A is decreased. So option A is the correct option. What is karyotype of Turner syndrome? So Turner syndrome, remember, it is monosomy X. One chromosome is less, so 45XO or 45X. That is your Turner syndrome. Remember the features of Turner syndrome. Klinefelter syndrome, what is the typical hormonal pattern? I already explained this to you. There is primary testicular failure. Whenever there is testicular failure, testosterone is going to be low. So normal testosterone is out of question. Raised testosterone, again out of question because there is primary testicular failure, so no testosterone is being formed. So whenever testosterone is raised, all these are out of question. Normal testosterone. So this, there is raised testosterone. We will see what happens to this. Normal testosterone, again, out of question. Decreased testosterone. Yes, because primary testicular failure leading to less testosterone. And what is going to happen? Here, though testosterone is decreased, FSH is less. Whenever testosterone is less, FSH and LH is going to be high. So this is the correct option. Low testosterone, high levels of FSH and LH, and in addition, again, high levels of SHBG, sex hormone binding globulin. If that is there on the option, even that is going to be raised. What is the recurrence risk for Down syndrome with parental Robertsonian translocation? Similar to general population, we saw that no, it is not similar to the general population. The risk is greater. So it is about 10 to 15% if the mother is the carrier. Okay. If the father is a carrier, then it is about 1 to 2%. But if it is maternal, the risk is definitely high. Prader-Willi is caused by, there is deletion. So C, D, and E, both of them are gone. Prader-Willi is because of paternal. Okay, so paternal 15q11 deletion, you will get Prader-Willi. When it is maternal, you will get Angelman. Huntington disease shows anticipation due to expansion of which trinucleotide? CAG, CGG, CTG, GA, or GCC. So it is, remember, it is CAG. Okay. Fragile X is because of expansion of CGG. Myotonic dystrophy is CTG, and Friedreich's is GAA. You have a pedigree which shows that the father is affected. Remember, affected father. All daughters are affected. All 4 daughters are affected, and 3 sons are unaffected. What is the most likely inheritance? So from father to daughter, and all daughters are affected. Definitely, it is not autosomal, because if it is autosomal, the sons are also going to be affected. And it is definitely not mitochondrial, because if it is mitochondrial, none of the children will be affected. So it is X-linked dominant, because all the daughters, they are going to get that affected X chromosome from the father. And if it was autosomal dominant, then it would be equal distribution between males and females. The males can also be affected. So you have another pedigree. The mother is affected. Father is unaffected. So affected mother, normal father. So you have this affected mother, normal father. All 4 children are affected. They are males, females, but so even males are affected, even females are affected. Because all are affected, I am putting it as solid. Now the affected son's children are all unaffected. Now the children from the son, they are all normal. Whether it is male or a female. What is happening here? The disease is being transmitted only from the mother to all the offspring, but from the father, it is not being transferred anywhere. This is very much characteristic of mitochondrial transmission. Okay, from the mother to all the children, but from the son, it will not go to any of them, because all the mitochondria in the sperm, they are deleted. All right. That brings us to the end, and let's begin the third part now, where you have disorders and prenatal. I will just get a glass of water. You have single gene disorders, cancer genetics, which is hot topic, prenatal screening and diagnosis, genetic counseling and risk calculations, and a few SBAs. So, single gene disorders, you have to know them in detail. Cystic fibrosis, which is autosomal recessive. The affected gene is gene CFTR gene on chromosome 7. And the commonest mutation that is seen in Caucasians is F508, delta F508. The carrier rate is 1 in 25 and affected individuals are 1 in 2,500. This is for Caucasians. Okay. In male, they present with infertility because congenital absence of the duct, CVAD as we call it, and there is azoospermia in the male. And the other organ systems like lung and pancreas, they are also affected. The diagnosis, if you do chlorides in the sweat and chloride is more than 60 millimoles per liter, that is diagnostic of cystic fibrosis. Now coming to sickle cell, this is another disorder which is autosomal recessive. Okay, and there is a defect in the beta globin chain. Defect is there or chromosome 11, where the amino acid glutamine is replaced by valine at position 6. So there is only substitution of one amino acid that gives rise to this disorder. The good thing is people who are carriers of sickle cell, they get protection from malaria. Because the malarial parasite, because the cells are smaller in size, the malarial parasite is not able to replicate in the cells. It is seen in about 1 in 10 to 12 black population. The trigger for sickle cell, we know that whenever there is hypoxemia, the cell, it becomes into an abnormal cell. It becomes sickle cell. Unlike the normal cell, which is round, the sickle cell, it becomes sickled. And then this sickle cell, it causes obstruction in the small vasculature. So the trigger for this sickling is hypoxia, dehydration, infection, or cold. Then other single gene disorder that you should be aware is thalassemia. So you have alpha or beta thalassemia. For alpha thalassemia, the defect is there in chromosome 16. For beta, it is chromosome 11. And there is defect in production of the globin chains. So whenever the beta chains production is defective, we call it as beta thalassemia. Whenever there is defect in production of alpha chains, it is called as alpha thalassemia. Now next coming to Marfan. So Marfan is autosomal dominant. There is a defect in formation of fibrillin, and that is why you get a defect in all the systems that are there. So these individuals are unusually tall for their age. There is arachnodactyly. They have pectum cavus, aortic root dilatation because the collagen is defective. So the vasculature, especially the aortic root, you get aneurysms and you get dilatation. They have hypermobile joints. There is subluxation of the lens. But their intellect is normal. Their mental capacity is normal for these individuals. What we have to remember as obstetricians is that females, if they are planning a pregnancy, if the aortic root is more than 4.5 centimeters, it is a contraindication to a pregnancy. And unless this aortic root replacement is done, you will advise them against planning a pregnancy. Now, Huntington's chorea is autosomal dominant. There is in chromosome number 4, there is repeat of this trinucleotide, CAG. Onset is normally there at about 30 to 50. We saw that in successive generations, the onset is going to come on earlier because from the parent to the offspring, this expansion of CAG, it goes on increasing. So the parent might show the symptoms if the parent is the carrier of this Huntington gene. For the parent, the onset will come on at around 50 years of age. In the offspring, it will come around a little bit earlier. say maybe 40 to 45 in the second generation. In the third generation, it will come on still earlier. And the severity of the disease, it goes on progressing as the generations pass. There is chorea, there is dementia, and it is 100% penetrance is seen. Now, fragile X is X-linked dominant, so only passed on usually from the mother. Okay, it is X-linked dominant. So, from the father, it will go only to the daughters. The expansion is there in CGG trinucleotide. It is the commonest inherited cause of intellectual disability. The important point to remember, it is the commonest cause of acquired cause or inherited cause of intellectual disability. Other features that are there, long face, large ears, macroorchidism, and post-puberty. This is not there at birth. After puberty, you will see macroorchidism. Duchenne's muscular dystrophy, again, it is X-linked recessive. There is a fault in dystrophin at chromosome 21, X part. So there is calf pseudo hypertrophy. Gower's sign is positive and the levels of creatine kinase are increased. So Gower's sign is because there is weakness in the muscles, especially weakness of the calf muscles. These individuals, they are not able to get up or stand up properly from a sitting position. So what they do is, if they have to stand up, especially in infants, they will roll over onto their stomach and they will make use of the hands and both the feet to get up. So that is called as the Gower's sign. Then von Willebrand's disease, autosomal dominant. It is the commonest inherited bleeding disorder. Again, von Willebrand's, there are different types. Some are recessive, but commonest type of inheritance is autosomal dominant. Then hemophilia A, it is X-linked recessive. There is deficiency of factor 8. The common finding there is bleeding, spontaneous bleeding into the joints. So the commonest hits that you have to recall is cystic fibrosis. It is autosomal recessive. Defect in CFTR. The commonest mutation that is seen is delta F508. Sickle cell, autosomal recessive. There is defect in the beta globin where glutamine gets replaced by valine. The amino acid glutamine is replaced by valine. Marfan's, it is again autosomal dominant. There is defect in FBN1 allele. Aortic dissection is a very common feature seen in people with Marfan. Others are unusually tall, subluxation of the lens, joint hypermobility. Then Huntington's, again autosomal dominant. The repeat is CAG, trinucleotide. Penetrance is 100%. In Fragile X, it is the commonest inherited disorder for intellectual disability. In Duchenne's muscular dystrophy, it is X-linked recessive. There is defect in dystrophin and Gower's sign is positive. Hemophilia A, X-linked recessive, deficiency of factor 8. Von Willebrand's is commonest inherited bleeding disorder. It is autosomal dominant. Tay-Sachs, autosomal recessive, commonly seen in Ashkenazi Jews. It is a type of inherited metabolic disorder. Then PKU or phenylketonuria, it is again autosomal recessive and you have a defect or absence of phenylalanine hydroxylase. Now coming to cancer genetics. So this is something which is commonly asked for you in the exams. You have to know what is Lynch, what is BRCA1, BRCA2, Li-Fraumeni, and other cancer syndromes. So Lynch syndrome, or also known as HNPCC, that is hereditary non-polyposis colorectal cancers. It is autosomal dominant. The MMR genes that are involved, most common is MLH1, MSH2, MSH6, and PMS2. Please remember this, it comes as an SBA. They will ask you which is the commonest MMR gene that is implicated in Lynch syndrome. Now, the marker for Lynch syndrome is this microsatellite instability. This is the one that is raised whenever the tumor testing is done. Okay. The commonest gynecological cancer that you see with Lynch syndrome is endometrial. It is about 40 to 60%. The lifetime risk in people, those who have Lynch syndrome, the risk of endometrial cancer is 40 to 60%. Other cancers that are common are colorectal cancers, 50 to 80%. Ovarian is a little bit less as compared to BRCA. And you can get gastric or urolethal cancers. Screening. So, because that would help to prevent. So, because gastric or colorectal cancers are common, you can offer them colonoscopy from the age of 25 and endometrial sampling from the age of 35. Aspirin is another drug that could be given to people with Lynch syndrome. It is said that it decreases the risk of colorectal cancers. Okay, so then you have BRCA. BRCA1, BRCA2. So remember, it is autosomal dominant. And there is a defect in the DNA repair genes, because we saw when we saw cell growth, or when we saw the cell cycle, the cell is always constantly monitoring to see whether there is a defect in the cell before the cell moves on to cell replication. Whenever this defect doesn't get corrected, or there is a defect in this gene, BRCA gene, so that is why there is a fault in the DNA and you get replication of the faulty cells that gives rise to cancer. So in BRCA1, there is a fault in chromosome 17. In BRCA, there is a fault in chromosome 13. Very common, this is very important for you to know from the exam point of view. For CA breast with BRCA1, the risk of CA breast is 70%. With BRCA2, it is 65%. For CA ovary with BRCA1, the risk is 44%. And with BRCA2, it is 17%. What I would remember from this is for BRCA1 or 2, risk of CA breast is higher than that of CA ovary. Whenever you get any ovarian cancer with BRCA, remember these are high-grade serous ovarian cancers, mostly serous ovarian cancers. The other types of ovarian cancers are not seen with BRCA. Okay. So that is, we have discussed now about Lynch. And we have discussed about BRCA. The other thing that we have to remember is that for the cancers, ovarian cancers, which are due to BRCA1 or BRCA2, PARP inhibitors are the ones which have been found to be very effective for chemotherapy. Okay, so these PARP inhibitors, they selectively act on this defective DNA that is there in the ovarian cancer cells, and the cells that have the normal DNA, those cells are not affected. Okay, so it's enough for you to remember that in any cancers due to BRCA1 or BRCA2, chemotherapy which is most effective is that with PARP inhibitors. If you remember that much, it's more than enough for you. Now, Lee-Fraumeni is another cancer syndrome where it is autosomal dominant. The gene is present on chromosome 15. It is PP53 gene that is defective. And you get cancers of, or more commonly, it is sarcoma. You get breast cancers, you get brain cancers, you get adrenocortical and leukemia. Very important, Lee-Fraumeni onset is again in childhood. So that is why the hypothesis, the two-hit hypothesis or the Knudson's hypothesis, which I already explained, that is applicable here. Okay, because both the alleles, they are inactivated. The affected allele is inherited and then the second hit appears very early in childhood. Other cancer syndromes that you have to be aware of, Cowden. So P10 affection is there. you get breast, thyroid, endometrial cancers, and also mucocutaneous hematomas. In Peutz Jeghers, there is mucocutaneous pigmentation. There are gastrointestinal hematomas, and ovarian tumors are also there. And the ovarian tumors that are there, these are sex cord tumors with annular tubules. That is very characteristic of Peutz Jeghers. Most characteristic is this mucocutaneous pigmentation that is there with Peutz Jeghers. Okay. All these cancer syndromes, remember, they are all autosomal dominant. And because of the Knudsen two-hit hypothesis, any cancers that present with familial cancer syndromes, they come on at an earlier age. So other things that you have to remember for those who have defect in BRCA1 and BRCA2, you can offer them risk-reducing bilateral salpingo oophorectomy. So what happens if once the family is complete and you do a salpingo oophorectomy, it reduces the risk of ovarian cancers by almost about 96%. There is a 4% chance of primary peritoneal cancers being present. So for BRCA1, risk-reducing BSO is offered at 35 to 40 years of age. For BRCA2, a little later on at 40 to 45. Cancers that are ovarian cancers with BRCA1, remember the chemotherapy of choice is PARP inhibitors. For Lee-Fromeny, the mutation that is there in P53, and you get sarcomas, you get breast cancers, brain cancers, adrenocortical tumors, and you get leukemia. Now, risk-reducing RRBSO is offered for BRCA1 at 35 to 40 years, for BRCA2 at 40 to 45. We already saw that you will give PARP inhibitors as chemotherapy. Okay, so I will not read all this because we have just read enough all of this. And please remember the two-hit hypothesis or the Knudsen's hypothesis. That is why all cancer syndromes that are there, familial cancer syndromes, the cancers, they come on at an early age. So in summary, Lynch, there is a defect in mismatch repair genes. Commonest is MLH1, MSH2, MSH6, and PMS2. Commonest cancer seen with Lynch syndrome, gynecological cancer, is endometrial, not ovarian. BRCA1, the defect is in chromosome 17. BRCA2, it is chromosome 13. The ovarian cancers that are there with BRCA, they are serous cancers, okay? High-grade serous cancers, the defect is in P53. For those who have BRCA1, risk-reducing bilateral salpingo oophorectomy offered at age 35 to 40. In Lee-Fromeny, the defect is in TP53. Cowden, the defect is called as P10 defect. Peutz Jeghers, the cancers that are seen are STK11. PARP inhibitors, the preferred agents of chemotherapy for ovarian cancer is PARP inhibitor in BRCA genes. And because of the two-hit hypothesis that both the alleles have to be inactivated for cancer to develop, because of that, in familial cancer syndromes, the cancers, they come on at an earlier age. Now coming to the last topic, that is prenatal screening and diagnosis. So, screening for Down's syndrome is the one that is commonly asked. Now in UK, screening for Down syndrome is offered universally. That means it is offered for all pregnant females. And you can either do it in the first trimester, or if it is missed in the first trimester, it can be done in the second trimester at 15 to 20 weeks. So in the first trimester, you can do the combined test, which is done between 11 to 13 weeks of pregnancy. Here, combined test means what is done? The nuchal translucency is measured because we know that in Down syndrome, if it is more than 3.5, it is diagnostic, or it says that it is a high risk. And in addition to nuchal translucency, the levels of Papa A and beta HCG are measured, and also the maternal age is seen, because as maternal age advances, we know that the risk of Down syndrome increases. So there are cutoffs that are available depending on the maternal age, and according to that, you can then predict whether this fetus or this pregnancy is at a high risk or it is at a low risk for Down syndrome. Okay. So the rate of detection for trisomy 21, it is about 85% with the combined test, with false positive of 5%. So we can say that it is fairly accurate, or it has a good predictive value for Down syndrome. So whenever you have Down syndrome, we saw that HCG is increased, HCG inhibin, but inhibin we do not do in the combined test. HCG is increased, Papa A is decreased, and nuchal translucency is also increased for Downs. Now for trisomy 13 and for 18, again nuchal translucency is increased, but HCG is decreased, and also Papa A is decreased. So please remember, for Downs, HCG is increased, whereas for trisomy 18 and 13, HCG is decreased. And Papa A is decreased for both. Nuchal translucency is again increased for both. Now, if any lady, she misses the first trimester screening, or the combined test is missed, you can do the quadruple test. It is done at 15 to 20 weeks of pregnancy. So here, 4 biochemical markers are there: alpha fetoprotein, HCG, estriol levels, and inhibin A. Detection rate is a little bit less, it is about 75%. So for trisomy 21 or for Down, HCG and inhibin are increased, alpha fetoprotein and estriol, they are decreased, and alpha fetoprotein is very low. Normally, alpha fetoprotein, we are looking out for neural tube defects, especially open neural tube defects. We know alpha fetoprotein is going to be raised. Whenever you see low alpha fetoprotein levels, think of Down syndrome. Now, I think I have that in the next slide. Let me just check. Okay, we will discuss in detail about non-invasive prenatal testing. So coming to the timeline. At 10 weeks, you will offer non-invasive testing, or you will do cell-free fetal DNA, because we know that from 10 weeks of pregnancy, little bit of material from the fetus, it escapes into the maternal circulation. And when you draw blood from the mother in the serum, you can get little bit of the fetal plasma, of the fetal DNA. That can be analyzed, and you can know whether there is any defect in the fetus. Before 10 weeks, the amount of fetal DNA is very less. So it is offered only after 10 weeks. The detection for trisomy 21 or Down is about 99%. It is very accurate, with false positive only 0.1. But again, important thing to remember that cell-free fetal DNA or NPT, it is not diagnostic. It is only a screening test. And if it shows that NIPT, there is a high risk of trisomy 21, you have to offer the mother a diagnostic test or an invasive test in the form of either an amniocentesis or a chorionic villus biopsy. So chorionic villus biopsy, it is done between 11 to 14 weeks of gestation. It can be done transabdominally or transcervical also. It is quicker because the karyotype that you get back, it comes back sooner. But remember, it is an invasive test. The risk of miscarriage, not 1%. It is now the newer guidelines say both for CVS and for amniocentesis, the risk of miscarriage is 0.5 to 1%. The danger or the drawback of CVS is that in sometimes you can get confined placental mosaicism in some of the fetus. So this mosaicism that is there, it is there only in the placenta. And if you are sampling that part of the placenta where the mosaicism is present. That is why you get this 1% false positive rate when you are doing a chorionic villus sampling. Amniocentesis, it will be done after 15 weeks of pregnancy because you need good amount of amniotic fluid. You have to collect the amniotic fluid and then that fluid is sent for karyotyping. So amniocentesis is always done under ultrasound guidance because you want to avoid the placenta. It is very good for definitive karyotyping, and because a lot of fluid is also available, biochemistry, a lot of biochemical tests also can be done on the amniotic fluid. Similar to CVS, in the best of hands, the risk of miscarriage is 0.5 to 1%. The results usually take a longer time to come, but the accuracy is better than that with CVS. Okay, so that is all about the invasive testing. Okay, so NIPT done at 10 weeks, then the combined test is done at 11 to 13 plus 6 weeks, where you will do nuchal translucency, Papa A, beta HCG, and you will also see the maternal age. So for Downs, NT is increased, HCG is increased, Papa A is low. It's important to remember the detection rate is about 85% with the combined test. False positive is 5%. Quadruple test is done at 15 to 20 weeks, normally not done. Only if the combined test is missed, then the quadruple test is done. So here, 4 biochemical markers are tested: alpha fetoprotein, HCG, estriol levels, and inhibin. So in trisomy 21, you will get too high and too low. HCG and inhibin are raised and the others are low in trisomy 21. Okay, the detection rate is usually about 25%. Please remember, if there is a neural tube defect, open neural tube defect, only then alpha fetoprotein will be raised. Otherwise, when you have low alpha fetoprotein levels, it is usually an indicator of Downs. If quadruple test is also missed, then you will do an anomaly scan. So 18 to 20 weeks anomaly, 18 to 22 weeks in the anomaly scan, you will have a structural survey. You will have a look at the heart, brain, spine, kidneys, limbs, and placenta and liquor. Some soft tissue markers are there seen in Downs. So hypotonia, or if there is hypertelorism that is seen, or the thumb that is adducted, then usually they say it is a marker, or sometimes duodenal atresia, cystic hygroma. All these are seen in Downs. So whenever you have any of these markers that are seen at the anomaly scan, you will refer them to the fetal medicine unit and the multidisciplinary team for a specialized scan again. So a little bit of detail again about this prenatal screening and diagnosis. So non-invasive prenatal testing, or NIPT, or cell-free fetal DNA, it is done, offered from about 10 weeks of gestation. At that time, the fetal fraction in the maternal circulation is about 5 to 10%. The diagnostic accuracy for Downs is 99%, false positive is 0.1%. Remember, it is a screening test, it is not diagnosis. You will confirm by some invasive tests like CVS or amniocentesis. So we have already seen about CVS and amniocentesis. Now, fetal blood sampling or cordocentesis, that means you put your needle directly into the cord and you obtain a sample of the fetal blood. These cordocentesis done in very specialized centers, it is done very rarely. It is done basically for diagnosis of fetal anemia, and also whenever you want to give intrauterine transfusion to the fetus, like parvovirus infection, where there is severe fetal anemia leading to hydrops, or any other condition wherever there is fetal anemia and you want to give fetal transfusion, that is the time cordocentesis is done. And pre-implantation genetic testing, or PGT, it is done for individuals, those who are undergoing IVF, especially if they have single gene or chromosomal defects. So once fertilization is achieved, one cell is removed, and then that cell is tested for this gene defect or the chromosomal defect, and embryos which are free from the effect, those are the embryos that are transferred back into the uterus. So remember the markers. So for in combined test, for these are markers especially for Down syndrome. HCG is increased, Papa A is decreased, nuchal translucency is increased. In quadruple test, HCG and inhibin are raised, alpha fetoprotein and urinary estriol are the ones that are decreased. Now, the recalls or important points that you have to remember is combined test done between 11 to 13 plus 6 weeks. For trisomy 21, HCG and NTA is raised, Papa A is decreased. With quadruple test, HCG is raised, inhibin is raised. So too high and too low. Alpha fetoprotein and urinary estriol, they are the ones that are low. Alpha fetoprotein is decreased in Down syndrome. Please remember, it is increased in neural tube defects, especially where you have open neural tube defects. Why? Because the cells, they are exposed to the amniotic fluid and they release a lot of this alpha fetoprotein into the amniotic fluid, which then gets picked up. Also, whenever you have cleft lip or cleft palate, even in those fetuses, you have raised levels of alpha fetoprotein. Or even in abdominal, like there is a gastroschisis, even in those, there is raised levels of alpha fetoprotein. Now, NIPT or cell-free fetal DNA used for diagnosis or for screening of Down syndrome, not diagnosis, screening. It is 99% positive or predictive for detection of Down syndrome. Remember, NIPT is a screening tool, it is not diagnostic. With CVS, which is done between 11 to 14 weeks, the rate of miscarriage, the newer guidelines say it is 0.5 to 1%. It is same both for CVS and for amniocentesis. Amniocentesis is done from 15 weeks onwards. Miscarriage is about 0.5 to 1%. The only drawback that is there with CVS is that if there is a placental mosaicism, sometimes when you are taking a sample from that localized part of the placenta, 1% of the times you can get a false positive rate result. And pre-implantation genetic diagnosis is done for IVF. So any of the embryos that have the known mutation, those embryos will not be transferred back into the uterus. Now, coming to the Hardy-Weinberg equilibrium. So you will not be usually given these type of problems in the exam. At least I have not seen, but you should be like knowing a little bit about that. So what is this Hardy-Weinberg equilibrium and equation? Okay. So this P plus Q, that is to know the allele frequencies. We know that for any gene, there are two alleles. So P and Q. So P is said to be the dominant allele and Q is said to be the recessive allele. Okay, usually you will not get both dominant. You will get one dominant. The alleles are one dominant and one recessive. So it is said that in a given population, this frequency or the allele frequency of P plus Q, it remains constant at 1. So if the quantity of the dominant allele increases or P increases, then Q will decrease to maintain this frequency, or if Q increases, then P will decrease. There are some conditions that have to be fulfilled before this equation is true. That means there are no inheritable influences that are there. Okay. So in any population, the population size has to be large. Then there is no random mating that is seen. There should be no migration. That means no outside individual will come and no individual from here will go outside. So these are a few criteria that have to be fulfilled before this equation is true. Okay, so no outside influences should be there. Now to know the genotype frequencies. So the equation that is there is P square plus 2PQ plus Q1. that should remain as 1. That should remain a constant. Okay. So here in this, again, we saw that P is the homozygous dominant. P square is homozygous dominant. 2PQ means a carrier. There is one dominant and there is one recessive allele, and Q2 is a homozygous recessive. So this equation also has to remain 1. Now this equation is useful or helpful to us to calculate the genotype frequencies. Okay. For an example, if you have cystic fibrosis in an Caucasian individual. So in Caucasians, we know that the frequency of cystic fibrosis is 1 in 2000. Now, cystic fibrosis is something which is a recessive gene. Okay. So it is autosomal recessive. Now, recessive means the individual has to be homozygous. So Q2 is 1 in 2500. So this is a square. What is the square root of 1 in 2500? It is 1 in 50. Okay. So Q is 1 in 50. Now, to calculate the disease frequency or to calculate the carrier frequency, if you see this equation, if you know this 2PQ, okay, so this 2PQ you will multiply by 1 in 50. So 2 into 1 into 1 in 50, that will give you 1 in 25, or that is the carrier frequency for the gene cystic fibrosis in Caucasians. So that is how this equation, it helps you to calculate the genotype frequencies for any given disease. Like even if you have one value is known to you, you can easily calculate all the other values. From an exam point of view, I have not seen any, but because the carrier frequency or what is the disease in the general population, all that will be given to you in the exam. Now, what is the risk to the offspring? It will be 1 in 4, because 4 offspring, hypothetically, from each parent, you can get 4 offspring. So 1 in 4 into P. So why P? Because P is dominant. Okay, P is the dominant allele, Q is the recessive. So if you want to find out whether the offspring is going to be affected or not, you have to know how many of the offspring are going to inherit the dominant gene. So if you want to find out the risk to the offspring, 1 by 4 into P. Okay, so that will give you what is the risk to the offspring. Now this Bayesian risk adjustment, so this is something which is it is a statistical thing. You need not know it in detail, but to calculate what is the risk of frequency in a general population, you will have to know what is the basic rate of any disease or the prior rate of any disease in a given population. How much is the conditional influence? That means outside influence that is affecting this disease. So this prior or the basic incidence, it is multiplied by whatever conditions are being imposed on that population. And then the risk at present is calculated. So that is called as the posterior risk. You need not go into details of that because I've not seen any questions like this in the exam. Now, we know that whenever there is any genetic disease in the family, so the couple or either of the parent, they will come to you for counseling, either periconceptionally, preconceptionally, or early on in pregnancy. So there are a few principles that you have to follow. The first is it should be non-directive. That means whatever options are there for that particular disease or for that particular condition, you will be telling the options to the individual or to the couple, and you will not impose your decisions onto the couple or to the individual. Second is always maintain confidentiality. But you will tell them what is the risk to the relatives, like BRCA1, BRCA2. We know that other members of the family could also be at risk of developing that cancer. So even though you are maintaining confidentiality of that individual or the couple, you should be aware of what are the risks to the other relatives. Now, to come to a diagnosis of the inheritance pattern, you should have the pedigree chart, which contains at least 3 generations. Only then, confidently, you can diagnose the risk, the type of inheritance pattern. Whenever the couple comes to you for reproductive options, you have to talk to them about pre-implantation genetic diagnosis. You can talk to them about donor gametes or donors, like donor ovum or donor sperm, then prenatal diagnosis and adoption. So prenatal diagnosis in the form of CVS or amniocentesis. Now, what are the recurrence risks that are there? So if you have autosomal recessive disorder and both the parents, they are carriers. So one out of 4 or 25% of the offspring are at a risk of getting that autosomal recessive disease. If it is autosomal dominant and one parent is affected, then 1 in 2 or 50% of the offspring will be affected. If it is an X-linked recessive condition and the mother is a carrier, okay, so 1 in 2 son will be affected, 1 in 2 daughters will be carriers, 1 in 2 son will be normal, 1 in 2 daughter will be normal. With Down syndrome, there is a 1% risk of recurrence, especially if there is a maternal translocation, okay, Robertsonian translocation, or the maternal age that is there. If the maternal age is very high, the risk of recurrence is increased. For translocation type, yes. So for translocation type, it is up to 100%, especially if the parent is an unbalanced carrier. If it is a balanced translocation, we saw in the previous question that especially in the mother, the risk of recurrence is about 10%. But if it is an unbalanced translocation in the parent, it is about 100% risk of recurrence. So coming to the recalls, Hardy-Weinberg, so to calculate what is the genotype frequencies, the expression or the equilibrium that is given is p square plus 2pq plus q square is equal to 1. To calculate the carrier frequency, you will multiply the square root of the disease frequency by 2 for autosomal recessive. Any autosomal recessive, if both the parents are carriers, 25% of the offspring is going to be affected. For autosomal dominance, 50% of the offspring is affected. Whenever it is an X-linked recessive disorder, 50% of the males are affected, or 50% of the male sons are affected. For T21 or Downs, sporadic recurrence is about 1%. If it is translocation in the parents, the risk is higher. It is much higher. Okay. Counseling, remember, it has to be non-directive. Whenever you are reading a pedigree or you are documenting a pedigree, in order to come to a conclusive or definitive diagnosis about the inheritance pattern, you need at least minimum of 3 generations that are there in the pedigree chart. And to calculate the Bayesian frequency, you will multiply prior and conditional, and that will give you the posterior risk or the frequency, the risk that is there at present. So let us see a few SBAs now. Most accurate test for definitive fetal karyotype at 16 weeks. So 16 weeks, definitely not CVS. It cannot be, and they are saying it is definitive, not screening. So it is not NIPT. It is not even quadruple test. CVS is out of question. Anomaly scan not done at 16 weeks. So you're left only with the option of amniocentesis. Why is my slide not going ahead? Sorry. Okay, so amniocentesis is the correct answer. Okay. NIPT or cell-free fetal DNA for T21. What is the key limitation? Okay. Detection rate less than 50%? No, detection rate we saw it is almost 99%. Cannot be done before 20 weeks. It's done at 10 weeks. Okay. Screening test only. It needs CVS and amnio for confirmation. It is correct. This is one correct option. Cannot detect T21? No, it is done very commonly for T21. It causes 1%. It is no, it's non-invasive, so it will definitely not cause miscarriage. So by default, it is only a screening test, and it needs amnio or CVS for confirmation. Now, though this NIPT, it has 99% specificity and only 0.1% false positive rate, there are a few limitations for this, that whenever there is obesity in the mother, the fetal fraction in maternal circulation is low. So you might not pick up in about 99% of the cases. In twins also, there is confusion, because especially if it is dizygotic twins, for monozygotic twins, it's not a problem. And whenever there is confined placental hemostasis, in those cases also, you cannot come to a diagnosis. Carrier frequency for cystic fibrosis in Caucasian populations. We know the carrier frequency, it is 1 in 25. Okay, so this you can calculate with the Hardy-Weinberg equation, and I already did that for you. So that is how you, but for this, you should, or they would give you what is the disease frequency in Caucasians. So when they give you the disease frequency, because and you also have to know what is the type of the disease, whether it is autosomal dominant or it is recessive. So because cystic fibrosis is recessive and Q is the one for the recessive gene. So here Q2 or the disease frequency is 1 in 2500. So Q2 is 1 in 2500. So Q becomes 1 in 50. And then that you can replace in this equation. So 2PQ is equal to 2 into 1 into 1 by 250. That is 1 in 25. Okay. Maternal alpha-fetoprotein is increased in which conditions? We saw whenever there is open neural tube defect. That is the only one. In Downs, in fact, it is decreased also. And also in abdominal wall defects, there is multiple pregnancy, yolk sac tumors. In all these also, alpha-fetoproteins are going to be raised. Which is X-linked to dominant condition? For this, it is a knowledge-based area. If you have not read, you will not know. So it is Rett syndrome. All the others, in fact, they are recessive ones. Okay, and they are X-linked recessive. Mitochondrial inheritance is there. The father has, you have an affected father and he has 4 children. How many of the offspring are at risk? It is mitochondrial inheritance. So none of the children are going to be at risk. Please remember, because all these affected mitochondria that are there in the father, all of them, they are going to be destroyed at the time of fertilization. The sperm doesn't contribute any mitochondria to the zygote. Okay, so none of the children are going to be affected. BRCA1 mutation carrier, what is the recommended age at which you will offer RRBSO? So for BRCA1, it is 35 to 40 years. Okay. And why bilateral sapphic oophorectomy is offered? So you will remove the ovary and the fallopian tube also, because it is seen usually that these high-grade ovarian tumors, they originate from the fimbria of the tube. So that is why the tube is also removed. And for BRCA2, it is offered at 40 to 45 years of age, because the onset of ovarian cancers is later in BRCA2. And for BRCA1 also, once family is complete, once only after childbearing is complete, only then you will offer salpingo-oophorectomy. PARP inhibitors are particularly effective in, they are effective in ovarian cancers, and those ovarian cancers that are because of BRCA mutation. So BRCA1 or BRCA2, that is the time when these PARP inhibitors are affected. Commonest single gene cause of inherited intellectual disability, it is Fragile X syndrome. Okay, please remember this. And they have asked inherited cause. In Down syndrome also, you will have intellectual disability. But Down syndrome is not something which is commonly inherited. Fragile X is more commonly inherited cause of intellectual disability. In genetic counseling, what is the principle that is most important to follow? So please remember that it should be non-directive. You will only present what are the options you will tell to the couple or to the individual, and whatever is the choice of the individual or the couple, you will respect that choice. You will not enforce your decisions onto the couple or to the individual. Okay, you will present all the information, what are the risks, what are the options, but you will always respect autonomy of the couple or to the individual. Okay, and also respect confidentiality, but keep in mind that in certain cancer syndromes or in certain, there is a risk to the relatives also. So these are the high yield recalls that are there in embryology and in genetics. We have discussed all of these. Does it make sense for me to just keep reading them? I will not do that. But these are about 60 topics or 60 points that we have discussed today. So this is all from me about embryology and genetics. I hope the discussion was helpful to all of you, and we are looking for a feedback from all of you. I'm sure all of you have put in a lot of hard work, and I'm sure all of you are going to rock in this exam. Keep studying, guys. Very few days remaining for the exam, and the more you practice in this limited time that is available, it's certainly going to be helpful for you. And keep attending these sessions, because you have realized right now how crisp and important the topics that we are discussing, and definitely it's going to be very helpful for you in the exam. Thank you so much for attending, and good luck to you, not only from me, from the entire team at Study Medic. You are all going to rock in the exam. Bye-bye and take care.